Not gonna lie. Ooh, ooh. It is now being live streamed. Okay. I was gonna comment that I really love the logo, all things, artsy and stuff like that. Okay, should I give it a couple more, Matt?
Yeah, we can wait another minute or two, I think.
Okay.
I think we normally expect a few more than this.
Mm-hmm.
Okay, you want to kick things off, and then as we're doing the intro, more people will trickle in.
Absolutely. Okie dokie. Welcome everyone to our May monthly meeting. Thank you all for coming. This is one of the 2 different meetings that the student board of EPAASM puts together. So let's just kick this off. Matt?
Yep. So I'd like to introduce the student chapter of EPAASM, co-presidents, myself and Kayla, the vice presidents and rising presidents after this event, Marissa and Jenna, the incoming vice president, Ben, secretary Caitlin, treasurer Zach, and our faculty advisor, Dr. Shin. Without the help of everybody in the student chapter, this event wouldn't be possible. So I'd like to extend great thanks to everybody involved in putting all of this together. And as our last event that we're hosting as co-presidents, I'd like to say it's been great working with everybody. And I'm excited to see Marissa and Jenna take over and all the great things that they're going to do in the year ahead. Okay, thanks.
Yeah. So thank you. So just as a quick thing to say, students, for those that are attending and those that will watch the recording after, just as a quick reminder, joining EPAASM is free. We as students really benefit from joining EPASM from a number of different ways. We obviously get to listen to a lot of wonderful speakers and meet a lot of individuals working in different branches of microbiology and immunology, but also we get to interact with each other and really network. And so we do more than just science. We really kind of grow together and we can do fun things. I know it's hard to imagine scientists and fun things, but some of the cool things that we do is science after hours or go to exhibits. And we can also give back to the local community by doing like judging for local science fairs. And so this is for all the universities within the Philadelphia area. And some of the examples of them are listed on the screen.
So if you'd like to learn more or also become a member, like Kayla said, it is free for students. Definitely visit us out at epaasm.org.
Yep. And as a quick reminder, there is a call for nominations for the next cycle. So please take some time and just click on that link or go to ASM in order to nominate an individual that you know for the ASM Award and the ASM Microbiome Data Prize honors.
And then we'd also like to end by congratulating our very own members who are elected to the American Academy of Microbiology Fellows class of 2022, Dr. Garth Ehrlich from Drexel University and Dr. June Zhu from the University of Pennsylvania. Fellows are selected through their demonstration of outstanding achievements in their area of expertise, strong spirits of service to advance microbial sciences, and sustained commitment to teaching and mentoring new leaders in the field. So congratulations to Dr. Ehrlich and Dr. Zhu. And with that, I think we will kick off our first speaker of the evening. So I'd like to welcome Elijah Davis from Drexel University, who will be presenting his work, Toll-like receptors dictate microglia responses to beta coronaviruses. Elijah, take us away.
Sure. Can everyone see my screen and hear me properly?
Yes.
Yeah, looks good.
Awesome. Well, one moment. Okay, well, good afternoon, everyone, and welcome to my presentation, Toll-like receptors dictate microglia response to neurotropic beta coronaviruses. As that lovely introduction by Matt stated, my name is Elijah Davis. I am a fourth-year PhD student in the lab with Dr. Sonia Navis-Martin here at Drexel's College of Medicine. And it is my pleasure to speak to you today just about some of our current research going on. One of the primary focuses of our lab is to investigate immune response to viral infections of the central nervous system. These known infections account for about 15 to 20% of diagnosed neurological disorders and include complications induced by Zika, rabies, and even HIV. They can present with a range of clinical manifestations such as gradual or sudden onset, acute or chronic inflammation, or chronic infection, or severity of inflammation, which can dictate or regulate host damage and destruction of tissues within the CNS. And if left untreated, these known infections can result in death or even long-lasting complications. And really, that's one of the main concerns of clinicians is this long-term brain dysfunction, which can result from either direct infection of brain tissues, encephalitis or meningitis, which is just inflammation of the brain or the surrounding regions, and shown up here. Cytokine storm, such as what's involved in SARS-CoV-2 infections, dysregulation of the autonomic nervous systems, and loss of oxygen, such as hypoxia. And so due to the global impact of viral infections, there's a need to viral neuroinfections, there's a need for relevant models of disease. And so mouse hepatitis virus, first isolated in 1949, has become well established as a model of neurovirulence. It is a lineage A beta coronavirus, and this lineage also includes the 2020 SARS-CoV-2 Wuhan strain. It's enveloped positive sense single-stranded RNA that infects mice using CCAN1 receptor. It induces clinically relevant diseases in vivo, such as respiratory disease, hepatic liver disease, and neurological disorders. So they really are an excellent tool to analyze coronavirus replication pathways and post-immune responses. And so we in the lab of Dr. Sonia Navas-Martin used two main strains of MHV with two very different presentations of disease in infected mice. The first being MHV-JHM, which I will always denote in this presentation as blue, and the second being MHV-859, which I'll always denote as being this nice fuchsia color. And so MHV-JHM is primarily neurotropic, infecting the brain, and is extremely neurovirulent, inducing severe inflammation. However, it's unable to infect the spine, lungs, or liver. MHV-859, on the other hand, is weakly neurovirulent and induces minimal inflammation, but is capable of infecting the brain, spine, liver, and lungs. In terms of neurological disease, MHV-JHM is highly fatal, inducing acute lethal encephalitis, and I'll also denote MHV-JHM with a little picture of a skull. And MHV-859 induces chronic demyelination, so mice are able to survive this, but mimics multiple sclerosis. And I'll always denote MHV-859 with a little neuron. And so there's long-standing evidence that marine coronaviruses can infect the CNS. And so coronavirologists are not too surprised to see neurological manifestations of disease in human coronavirus infection. And really what we're seeing now is a rise in neurological complications in COVID-19 patients. Roughly 80% of infected individuals present with some type of neurological disorder, whether it be delirium, encephalitis, meningitis, or even cerebral vascular disease. And these complications can continue months to years after viral clearance. And so despite the ongoing debate as to whether coronaviruses can penetrate the brain's defenses and actually establish infections, it's evident that we need to understand the central nervous system's immune response to coronavirus infections. And so when discussing immunity of the CNS, we have to examine the role of microglia. These are innate immune cells that are often called the gatekeepers of the CNS. These cells maintain homeostasis and also initiate and regulate powerful inflammatory responses during neural infectious disease, making them essential players in disease pathogenesis. Now, microglia actively survey the environment and respond to stimuli such as infection, neuroinflammation, or even traumatic CNS injury. And that allows them to transition from a quiescent resting state to a active state, which can either be pro-inflammatory, inducing pro-inflammatory cytokines such as TNF-alpha, IL-1 beta, IL-6, or an anti-inflammatory state, inducing inflammatory mediators such as IL-10. Now, this ability to transition between pro-inflammatory and anti-inflammatory states allows them to regulate disease pathogenesis and disease resolution. However, dysregulated microglia responses can actually function in exacerbating disease and worsening disease outcome. Now, we're seeing that microglia may play a role in coronavirus infection. And so this is data from a paper in which human ACE2 transgenic mice were infected with SARS-CoV-2, and they saw an increase in IL-6 and TNF-alpha positive microglia, suggesting that infection with this virus induced a pro-inflammatory cytokine response within the CNS. However, how microglia respond and sense coronaviruses remains poorly characterized. And so my research focuses on the role of toll-like receptors in microglia responses to infections. These TLRs are transmembrane proteins expressed by various cell types, including innate immune cells like microglia, either at their plasma membrane or within endosomes. And they serve as innate immune sensors that recognize pathogen-associated molecular patterns or damage induced by infection by these pathogens, which can lead to release of cellular factors that also activate toll-like receptors. Now, these toll-like receptors do have a degree of specificity, though they are classified as innate immune molecules. TLR2 and TLR4 can recognize bacterial components, with TLR4 recognizing LPS and TLR2 recognizing triacyl or diacyl lipopeptides from bacteria. And TLR3 and TLR7 are often considered antiviral toll-like receptors, with TLR3 recognizing double-stranded RNA and toll-like receptor 7 recognizing single-stranded RNA. Now, upon binding to their respective ligands, the TLRs will dimerize and then activate via phosphorylation. This will recruit their adapter molecules, either MyD88 or TRIF, which leads to a signaling cascade that induces pro-inflammatory cytokines, or in the case of TLR7 and TLR3, potent antiviral factors such as type 1 interferons, such as interferon alpha and interferon beta. Now, there is increasing evidence that these TLRs play a role in coronavirus infections as mediators of antiviral and intercellular immune signaling responses. Interestingly, there's data that suggests that TLR4 and TLR2, which primarily respond to bacterial constituents, also play a role in this antiviral response. However, how these TLRs are influencing microglia responses remains to be fully elucidated. And so that led us to ask two main questions. Do TLRs and their adapters regulate marine coronavirus replication within microglia? And which TLRs or TLR adapter pathways mediate microglia cytokine responses during marine coronavirus infection? And so to address this, we acquired immunocompetent microglia, or otherwise known as wild type, for this experiment. TLR adapter deficient microglia, so deficient in either MyD88 or deficient in TRIF, and then individual TLR adapter deficient microglia, so deficient in TLR2, TLR3, TLR4, or TLR7. Now, for the sake of this presentation and for the sake of time, we will only be discussing data regarding the TLR adapter deficient microglia. So these cells were infected with both either strains of MHV, the neurovirulent strain JHM, or the neuroattenuated strain A59. And then 18 hours post-infection, we collected the supernatant for viral replication via plaque assay. And we also collected the lysates to look for protein responses via Western blot. And so interestingly, what we saw was that in TRIF and MyD88 defective microglia, there is a defect in viral replication for both MHV A59 and MHV JHM. Now, this effect is more pronounced in the neurovirulent strain of MHV JHM. Now, this was surprising. This we did not expect, because TRIF belongs to the TLR3 antiviral pathway. And we expected that when you remove TRIF, the virus would be able to replicate really without any breaks, as the antiviral responses has been eliminated. However, we're seeing that TRIF is being utilized by MHV, by these marine coronaviruses, in a way that supports viral replication. And removal of TRIF is a detriment to viral replication. Secondly, we were very interested in the results of mild MyD88 deficient microglia, in which viral replication was drastically reduced, almost below the limit of detection for our plaque assay. And so we wanted to confirm this by looking at the expression of viral nucleocapsid. And so we see that in addition to the reduction of nucleocapsid in TRIF deficient microglia, we see a complete reduction of nucleocapsid when you have JHM infected mild MyD88 knockout microglia. So once we saw that there is impaired viral replication and impaired viral protein production in JHM-infected mild MyD88 deficient microglia, we also wondered were there any defects in viral genome replication as well. And just briefly, genomic replication for coronaviruses start with the release of the single-stranded RNA genome via an endocytosed virus. You'll have the translation of a viral polymerase, which then leads to the generation of both genomic RNA, which is then packaged into nascent virions, subgenomic RNA, which is then translated into viral proteins and then assembled into those nascent virions, and also the addition of double-stranded RNA intermediates. So by staining our infected cells with antibodies, fluorescent antibodies against double-stranded RNA, we can visualize the production of this dsRNA intermediate as a function of viral replication. And so we see that at 18 hours, while we can detect robust expression of double-stranded RNA and JHM infected wild-type microglia, there is a complete abrogation of double-stranded RNA in the double knockout mild MyD88 microglia, demonstrating that there is some type of impairment in viral replication in these cells. The loss of MyD88 makes these cells refractory, resistant to viral replication when it comes to MHV-JHM. And so having demonstrated that at 18 hours, there was a defect in double-stranded RNA production, we also wanted to determine if this impairment occurs early in viral infection as well. And so we see that indeed at 3 hours post-infection, there is a defect in viral replication in MyD88 knockout cells. And so what this suggests is that the loss of MyD88 either initiates an antiviral state at rest in these cells, or the loss of MyD88 upon infection with MHV-JHM severely dysregulates antiviral responses that leads to resistance to infection. And so in investigating the mechanism behind this observation, we found in the literature that MyD88 has been shown to actually mediate interferon-related responses by preventing the phosphorylation of IRF3 in a TLR3-dependent pathway. And so we believe that perhaps in the MyD88 knockout microglia, there is a upregulation of interferons leading to the induction of an antiviral state, or at least the secretion of antiviral factors. And so to test this, we treated these JHM-infected wild-type or mild MyD88 infected deficient microglia with neutralizing interferon beta to eliminate the interferon beta response pathways. And so what we see is that while there is a slight increase in viral replication in the MyD88 deficient microglia treated with neutralizing interferon beta, it does not restore viral replication to the extent of wild-type microglia, showing that while interferon beta may play a role in antiviral responses, it is not solely responsible for this refractory nature of MyD88 deficient microglia infected with MHV. And so we are currently interested in looking at the mechanisms behind this response, and we're currently in the lab looking at more intercellular signaling pathways to perhaps shed light on this phenomenon. And so in examining antiviral responses to MHV infection, we also wanted to investigate the intense production of pro-inflammatory cytokines seen in highly pathogenic human coronavirus infections, also called the cytokine storm. And so activation of microglia can elicit a pro-inflammatory cytokine response either by direct infection by viruses, or by peripheral infection, in which virus infection induces either host damage or the release of viral constituents that cross the blood-brain barrier and are recognized by microglia, which then activates them in turn. And so we wanted to examine the pro-inflammatory cytokine response in our TLR adapter deficient microglia upon infection with MHV. And so what we see is that in TRIF knockout microglia, there is a dramatic increase in IL-6, showing that TRIF is acting as a negative regulator of this pathway in response to coronavirus infection. Now, this is specific to IL-6, as in TNF alpha secretion seems to be dependent, at least partially, on TRIF expression. We also saw that in double knockout mild MyD88 deficient microglia, there was a complete reduction of IL-6 and TNF alpha, showing that this TLR adapter is essential for the pro-inflammatory cytokine response induced by MHV. And so after investigating these responses in a TLR adapter deficient microglia, we also wanted to investigate the effect of TLR4 stimulation, which will induce both a TRIF and MyD88 dependent pathway, on MHV replication. And so in wild-type microglia, we infected them with MHV-JHM, the neurovirulent strain, and then treated with TLR4 agonist LPS, or a TLR4 inhibitor TAC242. And so what we saw is that in JHM-infected microglia, there is a reduction of TRIF. Now remember, JHM is a more neurovirulent strain of MHV. It induces very high cytokine responses. We also see that it now has the ability to downregulate TRIF, which is a negative regulator of IL-6, potentially being the reason why JHM is able to induce such high factors or high inflammation within the CNS. We also see that upon stimulation with LPS or inhibition with TAC242, this inhibition of TRIF is completely reversed. And this reversal also correlates with a reduction in nucleocapsid. And so we know that TRIF, as shown in this pathway on your left, can respond by inducing both interferon beta and interferon alpha, which are potent antiviral responders. And so by utilizing LPS, we can see that, that LPS is actually able to induce or reduce nucleocapsid expression, as well as loss of TLR4. So why is this? Why can the treatment of LPS reduce nucleocapsid expression in MHV-infected microglia? And so looking upstream of TRIF at TLR3, we actually see that in TLR4 inhibited microglia, there is a upregulation of TLR3 phosphorylation and also an upregulation of interferon beta. And so this may explain why we have reduced viral nucleocapsid expression of a component of viral infection, of viral replication in TLR4 inhibited microglia. We also see a upregulation of TLR3 phosphorylation in LPS-treated microglia. And in fact, this upregulation of TLR3, which is induced by LPS, is TLR4 dependent. So why is TLR4 mediating TLR3 dependent TLR3 phosphorylation? This is a crosstalk pathway that has yet to be elucidated in the literature, and it's something that has not been described before. And so in conclusion, we see that loss of TLR adapters, MyD88 or TRIF, leads to a reduction in viral replication for both neuroattenuated strains of MHV A59 or the neurovirulent MHV strain JHM, with this effect being more pronounced in JHM and pronounced in the loss of MyD88, showing that or suggesting that MyD88 can regulate antiviral responses in the context of coronavirus infection. We also see that TRIF is a negative regulator of IL-6 in the context of coronavirus infection, but is partially essential or partially plays a partial role in TNF-alpha induced responses. We also see that mild MyD88 is severely needed, or loss of MyD88 is severely downregulates IL-6 and TNF-alpha responses in the context of coronavirus infection. And perhaps the regulation of these TLR adapters may play a role in the cytokine response or the intense cytokine response we see in clinical infections by human coronaviruses. And then lastly, we see that there exists this TLR4, TLR3 crosstalk pathway that might regulate interferon beta production and antiviral responses to MHV-JHM or MHV-A59. And so we're currently interested in analyzing both these MyD88 regulated antiviral responses and this TLR3, TLR4 crosstalk pathway in microglia. And so I want to conclude there. I want to first acknowledge my committee who's helped me so much on this pathway. Also the members of my lab, those that are both past, present, and new. And then I also want to recognize and plug, really, to be honest, these underrepresented minority support groups that really have enhanced my experience here at Drexel, such as the Bridge to Doctorate Fellowship, the Black Graduate Student Union, and of course, the Black Microbiologists Association. So I want to thank you for listening to that brief talk about why or how, what we're doing in the Novice Marlin Lab, and I would be happy to take any questions that you may have.
Great, thank you. That was a great talk, Eli. So for any questions for Eli, please type them in the chat and I can relay them to Eli. But I can also get us started. So I guess one question that I had is, what do you think the kinetics of this is? So you were working with knockout cell lines where you had knocked out the TLR signaling, but do you think this is something also where if you have infection and then you dampen the signaling of TLRs, you would see a similar response as well in dampening the inflammation that you're seeing? Or what are your thoughts around that?
So we're actually doing kinetic experiments right now, treating or infecting microglia with MHGJHM or A59 for early infection time points or late infection time points, and then suppressing the toll-like receptor response. And I believe you do still see this reduction of cytokine responses in these cells. Now, we haven't looked at plaque assay formation, or in fact, excuse me, let me stop sharing my screen. Yeah, so we haven't seen difference, we haven't analyzed plaque assay formation, but we still see a reduction in IL-6, TNF alpha, and IL-1 beta in these TLR inhibited microglia. And interestingly, it's not just the viral responding TLR, so TLR3 or TLR7, we're actually seeing a reduction in viral responses for TLR2 and TLR4 as well, which normally are not canonically known to respond to viral infections or coronavirus infections.
Great, interesting. Okay, so it looks like we have a question from Dr. Shin. So she says, great talk. Do you think that there is a soluble factor like a cytokine responsible for the requirement of MyD88, or is it an intracellular factor? And then have you tried transferring supernatants from wild type cells to MyD88 knockout cells to see if it rescues viral replication, or alternatively, if MyD88 knockout cell supernatant can suppress viral replication in wild type cells?
Oh, wow. I love the way you think, because we did that experiment as well. I do believe, or I guess I'll show my hand a little bit and say I did believe that there's some type of antiviral secreted factor in the MyD88 knockout microglia that is suppressing viral replication. So I did that experiment in which I took supernatant from MyD88 deficient microglia infected with JHM, and then treated wild type microglia with that supernatant, and then infected with my MHV A59 and MHV JHM. And there was no difference in the plaque assay numbers or viral replication in wild type cells treated with MyD88 supernatant, MyD88 deficient cell supernatant in the context of coronavirus infection. So that means that perhaps there's not a secreted viral factor being at work, but perhaps a antiviral state such as upregulation of helicases such as RIG-I or NDA5, which also play a role in coronavirus infection, or some other type of intercellular mediator of the viral responses. I have not tried transferring supernatant from wild type microglia to the MyD88 deficient microglia and infected that. That's a good point that I actually want to make note of and try that perhaps this week as well. So thank you for that question. That was a wonderful question.
Great, thank you. And then we have another one from Dr. Mel. So he's asking, what is the function of the crosstalk in this pathway in the context of microglia? Like, why would it be beneficial to induce antiviral responses when detecting bacterial LPS, if I understood correctly?
So this is very interesting. So I would say that we need to further elucidate the actual mechanisms of this pathway to see. Right now, we've only looked at TLR3 and TRIP, but I also want to look at the cytokines transcription factors such as IRF3, NF-kappa B, to see if there's any point or any alterations to these transcription factors, which could better shed light on why a crosstalk between TLR3 and TLR4 may exist, or the benefits of that. Now, I will say that we see that TLR3, TLR4 crosstalk pathway, but in the individual TLR knockouts, there is wide-scale reduction of pro-inflammatory and cytokine responses. And so it might not just be TLR3 and TLR4 that are responding to one another. There may actually be a TLR kind of signaling complex in which they're using MyD88 and they're also using TRIF. So there might be shuttling back and forth between the various TLRs that's inducing that crosstalk or inducing that responses. But no, we still need to dig deeper into the actual pathway of the TLR3, TLR4 signaling cascade.
And then we'll cap it with one last question from Dr. Schifferly. Are some of the detected MyD88 effects microbiome presence dependent, i.e., do you expect the same result in germ-free mice?
So this is an interesting question. So I'm sorry, let me clarify a previous point that the cells that we use are acquired from the NIH repository. These are cell lines. We did not immortalize cell lines or cell lines that we treated with a TLR inhibitor. So we did not derive them from mice. Now, I do believe that the microbiome does play a significant role in these TLR mediated pathways, especially in terms of leaky guts or dysregulated blood-brain barrier integrity, which could allow for trickling in of LPS or other bacterial constituents. So I do believe we need to repeat these experiments in vivo, as well as using TLR adapter or TLR knockout mice. So I do believe we would see a difference depending on the microbiome of these infected animals.
Great, thank you, Eli. Wonderful talk.
Thank you.
And with that, we'll move on to our next presenter.
Yep, great. Hi, everyone. So I'm going to introduce our next speaker. So our next trainee speaker will be Caitlin Grando, who is actually also our student chapter secretary. Caitlin is a graduate student at Temple University in Dr. Chala Tupul's lab, and today she will be presenting some of her work on the role of bacterial amyloid curly in salmonella-induced reactive arthritis. Take it away, Caitlin.
All right, thank you. As Marissa said, I'm a third-year graduate student in the lab of Dr. Chala Tupul at Temple University, and I'll be presenting my work on salmonella amyloid curly and reactive arthritis. So here, let me adjust this actually. So I'm talking today about Salmonella enterica cerevisiae. So this is a non-typhoidal strain of Salmonella, which means it causes gastroenteritis rather than typhoid fever. And of interest to us is that about 5 to 10% of these Salmonella infections progress to an inflammatory disease called reactive arthritis, which is an autoimmune disease associated with a genitourinary or GI tract infection about 2 to 4 weeks prior. Reactive arthritis is on the spondyloarthritis spectrum, and it causes swollen and painful joints and urinary tract inflammation. Reactive arthritis and spondyloarthritis are all highly associated with the HLA-B27 genotype, which is present in about 60 to 90% of cases. So because reactive arthritis is associated with infection, we are looking at bacterial products that could trigger autoimmune disease. And of interest to us is curly. So Salmonella and some other enteric bacteria form biofilms, which are multicellular aggregates that provide protection against antibiotics or the host immune system and secrete this extracellular matrix that is composed of polysaccharide like cellulose, extracellular DNA, and a bacterial amyloid like curly in the case of E. coli and Salmonella. So curly is a good candidate for a molecule that triggers autoimmunity because curly is a very strong immunostimulatory molecule. So when curly escapes the gut, it interacts with systemic macrophages. So in the extracellular matrix, curly forms complexes with this extracellular DNA. And then this curly DNA complex interacts with TLR2 via the amyloid component curly. The macrophage internalizes the curly DNA complex, and intracellular TLR9 recognizes the DNA component, and all of this upregulates pro-inflammatory cytokines and type 1 interferons. So our lab previously showed that infection with wild-type Salmonella and not the curly mutant, the CSGBA, induced anti-double-stranded DNA autoantibodies, which are a marker of autoimmunity common in diseases like lupus, though they're not necessarily associated with reactive arthritis specifically. The infection with wild-type Salmonella versus the curly mutant also induced elevated knee inflammation, as shown by synovial hyperplasia, which is a thickening of the cells in the membrane of the knee joint. So as I mentioned earlier, HLA-B27 is prevalent in about 60 to 90% of cases of reactive arthritis, while only about 4 to 8% of the population in the United States. HLA-B27 is an MHC class 1 molecule. That means it is expressed on all nucleated cells. And it composes the heavy chain, which binds to the peptide being presented, and beta-2 microglobulin. So there are two major hypotheses about what makes HLA-B27 pathogenic. The first is the arthrodogenic peptide hypothesis. So a distinguishing feature of B27 is that it has the cysteine 67 that's in the peptide binding pocket of the protein. So one idea is that B27 may just preferentially bind to foreign peptides that mimic self-peptides, allowing cross-reactivity of CD8 T cells to self-peptides. Second hypothesis is the misfolded protein hypothesis, which relies on a more unique property of HLA-B27. So that same cysteine 67 is able to form a disulfide bond and a homodimer of the HLA-B27 heavy chains. So these homodimers can, with no peptide being presented and no beta-2 microglobulin, be expressed on the cell surface and recognized by receptors on NK cells and CD4 T cells. This misfolded B27 dimer can also accumulate in the ER and induce the unfolded protein response, which induces interferon beta, IL-23, and activates the Th17 response, which we are particularly interested in because there is a known link between a Th17 response and reactive arthritis and autoimmune disease. We've also found in previous work in our lab that Curly also increases IL-17. So maybe the idea is that Curly could be synergizing with this unfolded protein response induced by HLA-B27 and triggering autoimmune disease. So the aim of my project was to determine how Curly is interacting with HLA-B27 genotype to induce autoimmunity. So the hypothesis is that Curly stimulates toll-like receptors, which together with the misfolded B27-induced UPR activation exacerbates the autoimmune response, including a Th17 response. I'm also looking at the effect of microbiota. So an inflammatory microbiota could further trigger and enhance the autoimmune response. So the first experiment I did was basically just to characterize the autoimmune response to Curly in HLA-B27 transgenic mice. Just basically to establish what kind of autoimmune response we see in this mouse model. So to do that, I IP injected Curly DNA that was purified from Salmonella biofilms, or IP infected the mice with Salmonella SpyB mutant. So that's a type B secretion system 2 mutant, which attenuates this strain so that it does not kill these mice because they're on a black 6 background, which are very susceptible to Salmonella infection. So it does still produce the autoimmune response without killing the mice over the course of 9 weeks. So I performed this chronic infection or injection, collected serum to look at autoantibodies, knees to look at knee inflammation, and then spleen and kidney to look at the systemic infection. So for the chronic infection, it looks like these mice maintain the systemic infection over all 9 weeks. And when we look at the autoantibodies, the anti-double-stranded DNA, the systemic infection with our SpyB attenuated mutant triggered autoantibodies by week 2, and then purified Curly injections triggered autoantibodies by week 5. Now looking at knee inflammation, it looks like only chronic infection induced a low-level knee inflammation, keeping in mind that these scores are out of 3, although it was significantly higher than our controls. But all of these levels of autoantibodies and knee inflammation are very similar to levels we've previously seen in wild-type mice. So there was no obvious exacerbated autoimmune phenotype, which is not entirely surprising, given that we did know these mice didn't develop spontaneous disease. So the second experiment is looking at how an inflammatory microbiota may affect this Salmonella-induced autoimmunity. So literature suggests that HLA-B27 transgenic rodents only with conventional and not specific pathogen-free microbiota can get arthritis. So what we used as kind of a model for this was segmented filamentous bacteria positive microbiota from mice that we got from Taconic farms. They're just known to be colonized with these bacteria. And SFB are known to elevate the Th17 response, which, what I said earlier, that's associated with reactive arthritis. And our preliminary data showed that SFB positive microbiota may increase curly-induced autoimmunity that we're seeing. So we did a fecal microbiota transfer from mice from Taconic into our B27 transgenic mice. Following the transfer, we did a streptomycin pretreatment. So this is not enough to strip the microbiota, but it does induce a low level of gut inflammation, creating an environment in the gut where Salmonella thrives and promotes a gastroenteritis rather than like a typhoid fever when they are infected with Salmonella. So following strep pretreatment, I infected these mice with wild-type Salmonella or a curly mutant Salmonella or LB as just a negative control. Because I was using wild-type Salmonella this time rather than my attenuated strain, this was only an acute infection over the course of 48 hours. And in this one, I'm directly comparing our B27 transgenic mice and Black 6 mice in parallel. So for this acute infection, we saw lower colonization by a little bit in our B27 transgenic mice after 48 hours. It wasn't by a whole lot. And then doing ELISA on the 48-hour serum, we saw a little bit of an elevation in the IL-23 response, which is interesting, specifically in the wild-type Salmonella, that's IR715 infected mice in HLA-B27 versus the Black 6 mice. This is interesting because, as I showed earlier, IL-23 can be induced by the unfolded protein response and activates Th17 cells. We also saw elevated GM-CSF in our B27 mice versus the Black 6 that were infected with wild-type Salmonella. And GM-CSF can be associated with autoimmunity. So next, looking at the unfolded protein response. So these three, HSP5 alpha, CHOP, and XBP1, are markers of an activated unfolded protein response. So we didn't see very much difference at all in HSP5 alpha, but CHOP was elevated overall, regardless of infection, in the HLA-B27 mice, and then XBP1 was elevated overall in HLA-B27 and specifically with the wild-type infection. So there was a very slight elevation in the unfolded protein response, I would say, in our B27 mice with the infection. Keeping in mind, these are coming from the cecum. So this is the site of the infection, but this is a mixed cell type tissue homogenate. So even seeing this low, the small amount of increase in the UPR was promising in vivo. So given the complexity of factors that were involved in vivo models, I also wanted to look in vitro for a more controlled environment to really look at the unfolded protein response, HLA-B27, and curly, and how they might be interacting. So to do this, I was using a BMDM model, so it's bone marrow-derived macrophages. So while unfolded protein response to misfolded B27 could occur in any cell type that expresses MHC class I molecules, so that could be any nucleated cell, I'd looked in BMDMs because we have a known response in these cells to curly, and we hypothesized that curly stimulation of the toll-like receptors and induction of the pro-inflammatory response synergizes and exacerbates this unfolded protein response to HLA-B27, which would be especially relevant in immune cells like macrophages, whose activation could provoke further inflammation. So we treated these BMDMs that we took from either our B27 transgenic mice or our Black 6 mice with curly, LPS, or ODN 1668, which is a TLR9 ligand, PAM3CSK4, which is a TLR2 ligand, or non-treated. Just remembering that curly activates both TLR9 and TLR2. So in these cells, we performed an MSD, which is just a multiplex ELISA, on the supernatants that we took from these cell cultures after 24 hours of stimulation with Curly, LPS, ODN, or PAM. And we saw a significant elevation of IL-6, TNF-alpha, and GM-CSF, which are all pro-inflammatory and elevated in the HLA-B27 cells in the pink in response to Curly especially. IL-1 beta was not much different between BLAK6 and HLA-B27. That is cytokine induced by the inflammasome. IL-10 is an anti-inflammatory cytokine, which was actually elevated in BLAK6, so kind of the opposite pattern. IL-27, IL-30 was elevated in B27 as well. IL-23, which is the one I talked about before, activates Th17 cells. It was not significantly different, but trended towards a little bit elevated, though overall the levels were pretty low in response. So this is also very interesting. So we looked at unfolded protein response markers in these cells as well. So this was qPCR on those same cells that we took the supernatants from. And all three of these markers of the unfolded protein response were elevated in specifically the stimulated B27 cells. So especially with Curly treatment and with LPS treatment. While if you look at the BLAK6 cells, there was pretty much no induction of the unfolded protein response at all. So this indicates that there is some crosstalk between this, probably the TLR stimulation of Curly and the unfolded protein response, but specifically in the presence of HLA-B27 and not in wild type cells. So in summary, in vivo, the B27 transgenic mice had similar knee inflammation, autoantibody production after SPI-B infection or Curly injection compared to BLAK6 in past experiments. If we looked at SFB positive, more inflammatory microbiota in these B27 transgenic mice, there were slightly elevated unfolded protein response and cytokine production compared to BLAK6 mice after an acute infection. And in vitro, the B27 transgenic BMDM secreted significantly elevated pro-inflammatory cytokines and elevated unfolded protein response activation after Curly stimulation compared to the BLAK6 BMDMs. So this ties into our kind of overall model that Salmonella infects the gut, invades and provokes this gut inflammation along with inflammatory microbiota or any other factors that would basically increase the leakiness of the gut and allow Curly that dislodges from biofilms in the lumen of the gut to leave the gut lumen. When this happens, Curly stimulates TLRs and together with HLA-B27 induced UPR activation, stimulates a strong systemic inflammation and especially a Th17 response, which we think is leading to arthritis. So the next steps with this project, I'm still pretty early days on it, is just to infect these B27 transgenic mice versus BLAK6 with my attenuated strain, so a chronic infection compared to a Curly mutant and SPI-B double mutant, which I'm in the process of making right now. So I want to be able to show the role of Curly during a chronic infection because previously I just did with the SPI-B infection. And especially because Curly alone wasn't provoking that much of a response, in these mice at least. I also want to include in that chronic infection the effect of a SFB positive microbiota and strep pretreatment to induce that gastroenteritis. I also want to repeat my BMDM experiments and look further if the UPR is continuing to be exacerbated in HLA-B27, look at some ER stress inhibitors, TLR antagonists to kind of establish the mechanism. Is it the TLR stimulation that is causing this, or is it some other mechanism? And I'd like to acknowledge everyone in my lab and my committee, as well as the histopathology facility at Fox Chase, who help us with scoring the knees for inflammation, and Dr. Sebastian Winter at Southwestern, who provided the SPI-B mutant salmonella that we used. And I can take any questions.
Great job, Caitlin. I'll clap on behalf of everyone. That was a wonderful talk.
Thank you.
So we do have a question in the chat. So Sonia said, great talk. She asked, is this pathotype observed in human pediatric population HLA B27 positive?
So I actually don't know a whole lot about pediatric population. I would say, so reactive arthritis, as far as I'm aware, tends to be more older people. And I know B27 alone is not typically just causing reactive arthritis because it's not uncommon in the population to have HLA-B27. So there's other factors involved, but I'm sorry, I don't know a whole lot about the pediatric population. That's interesting to look at though.
Great, thanks. And then Dr. Kutzler also asked a question. She said, if you could speculate a mechanism, do you think that if the TLR antagonists are mediating the effect? If so, what does this mean for translating the findings to individuals who are HLA B27 positive?
So that's an interesting question. So I think the idea for, so what I think is happening possibly is that there's literature showing that TLR stimulation, so unfolded protein response alone to just HLA-B27 isn't enough to provoke a very strong inflammatory effect. It's kind of a low level inflammation. But from what I can tell, it looks like it is kind of priming the immune response to have a stronger response to anything that activates TLRs, which currently we know is a very strong TLR activator. So that's kind of what I think is happening to really increase this immune response after like an infection. For translating it to individuals, I would say a lot of what is kind of the goal is to show that bacterial amyloids like Curly are playing this huge role in autoimmune disease. So if we can make therapies that target bacterial amyloids specifically, that would be really good. And that might mediate this effect. I know we're looking at like antibodies against bacterial amyloids. I'm sorry, that's probably, I don't know if I answered that question enough, but.
No, great. Thanks, Caitlin. And then maybe for sake of time, if it's okay, do you mind? So Sunny asked a great question. Do you mind answering her question in the chat, and then we'll move on to our final trainee speaker. Okay, great job again, clapping on behalf of everyone.
Great. So our final trainee speaker of the evening is going to be Nina Montoya. So Nina is a graduate student at the University of Pennsylvania in the lab of Dr. Joe St. Jem. And so today she will be presenting some of her work on the role of Kingella-Kingay lipopolysaccharide in galactan surface anchoring. So take it away, Nina.
Thank you, Jenna, for that introduction. So as Jenna said, I'm a fifth-year PhD candidate at the University of Pennsylvania, and today I'm going to tell you a story about the role of the Kingella-Kingay LPS, or lipopolysaccharide, in galactan surface anchoring. So I just want to quickly introduce you to our favorite pathogen in the St. Jem lab. So we work on Kingella-Kingay, which is an emerging pediatric pathogen. It's a gram-negative organism that's typically carried as a commensal of the oropharynx in young children. The oropharynx is this posterior region of the upper nose and throat. We also know from some recent molecular diagnostics that it is the leading cause of bone and joint infections in children between 6 months and 4 years of age. Furthermore, about 75% of the kids who end up developing clinical disease are under 18 months, suggesting that Kingella has this really limited or age-limited niche for infection, which we find quite interesting. We also know that while there are several clonal groups of Kingella, disease is associated with only a few of those, suggesting that there are some really specific molecular factors that drive pathogenesis. There's been a lot of really great work done both by our lab and by others that have helped us construct this picture of some of the really crucial steps in pathogenesis. So we know that Kingella-Kingay pathogenesis begins with colonization of the oropharynx, which is mediated in part by type 4 pili and their associated adhesive proteins, as well as some other adhesins that are expressed on the bacterial surface. And we know that following adherence to the respiratory epithelium, Kingella is able to invade the bloodstream through a breach in that epithelium. And once in the bloodstream, Kingella has to survive the trip to distal sites of infection, and this is specifically requires evasion of the host immune system. And when it reaches these distal sites of infection, Kingella kingae is able to cause inflammation and tissue damage, which is, the resulting infection. Excuse me. So I mentioned that intravascular survival is a really crucial part of Kingella kingae pathogenesis. And a previous grad student in our lab did an amazing job characterizing the mechanisms of innate immune evasion, specifically those required for complement resistance. So we know that Kingella expresses both a membrane-anchored polysaccharide capsule as well as an exopolysaccharide called the galactan. And what this previous student was able to show is that each of these surface polysaccharides are individually sufficient to prevent opsonine deposition and complement-mediated killing. And this was actually a really interesting observation because bacterial exopolysaccharides like the galactan are typically not implicated in processes like complement evasion, and especially not in such a prominent role. And so this really started to suggest to us that this galactan might be uniquely functional and have sort of an interesting way that it works during pathogenesis, and it prompted some further investigation of this structure. Additionally, while we know that the galactan is associated with the bacterial membrane, there's always been a question as to how it stays associated. So a little bit more about this galactan. This is a galactofuranose homopolymer exopolysaccharide. I'm showing you here the structure on the left. And in addition to its role in complement resistance, a previous student was also able to show that this galactan is also really good at promoting neutrophil evasion as well as evasion of antimicrobial peptide killing, again, highlighting this really multifunctional nature. There has also been some work done to understand the genetic basis of this polysaccharide. We identified this 5-gene locus called the PAM locus, and along the way, a group was able to show that the genes PAM, A, B, and C are sufficient for galactan biosynthesis. Naturally, that left some questions about the functions of these two downstream genes, PAM-D and PAM-E. So in my work, what I did was took a homology-based approach, both by sequence and by structure, to show that PAM-D has homology to a GTB superfamily glycosyltransferase, and PAM-E has homology to a family 25 glycosyltransferase, both of which have been implicated in LPS biosynthesis, suggesting that there might be a relationship between the galactan and the LPS. I also want to highlight that this type of genetic proximity, this cluster organization, is a bit of a rarity in Kingella. We typically find that genes that are co-transcribed in other organisms are scattered throughout the genome. So when we see this type of proximity, it suggests to us that there's a really crucial relationship between these genes and their respective products. So taken together, the sort of unique multifunctionality of the galactan, as well as the genetic proximity of the PAM genes, we hypothesized that the PAM-D and PAM-E genes are LPS glycosyltransferases, and furthermore, that they may facilitate a relationship between the LPS and the galactan. So to start interrogating this question, we started by isolating galactan from the bacterial surface, visualizing this material by Doc Page electrophoresis, and then staining it with silver. And then to confirm that any material that we recovered was in fact galactan, we performed a Western blot using a galactan-specific antiserum called GP19. And so I want to quickly introduce you to my strain panel before I get into these experiments. So in all of these experiments, our control or wild type strain is going to be this KK01 CSAA strain. And this strain is a capsule deficient strain, and we do that to avoid any contamination of the capsule in our various polysaccharide preps. So everything is going to be compared to this control strain. So what you'll notice in this experiment is that we were able to recover galactan only from the surface of the control strain. And that suggested to us that the genes PAM, D, and E are required for that recovery, suggesting that they might have a role in anchoring the galactan to the bacterial surface. So now, knowing that LPS or PAM-D&E are putative LPS genes, we wanted to characterize the Kingella kingae LPS since that had not been done yet. So in general, LPS is a tripartite molecule composed of lipid A, the core oligosaccharide, and then in some cases, a really long O antigen made up of repeating sugar units. So when you look at these LPS profiles on a gel, you can typically infer some information about these components based on the banding pattern. So if you look at something like salmonella, which we know has a really long O antigen, you see this really nice ladder pattern. So when we looked at our wild type or control strain Kingella kingae LPS, we found that our LPS separates into two very distinct modal clusters. So we found this low molecular weight, or LMW LPS cluster, that migrates with this really beautiful ladder pattern, suggesting the presence of some type of repeating unit. So we refer to this cluster as our atypical O antigen containing LPS. We also noticed that this high molecular weight, or HMW cluster, runs at a really larger size, suggesting that it has an additional polysaccharide component that is accounting for this difference in size. We also noticed that these LPS species are influenced by expression of the PAM genes. So in our PAM A, B, and C mutants, we lost this HMW cluster. And in our PAM D and E mutants, we lost both the HMW as well as the LMW cluster. And instead, we have this truncated form of LPS, suggesting that PAM D and E are required for the production of this atypical O antigen, and furthermore, that a full PAM locus is required for the production of this HMW LPS, implying that that HMW LPS species might contain both atypical O antigen as well as galactan. So in collaboration with the Complex Carbohydrate Research Center at the University of Georgia, we were able to do a compositional analysis of our LPS. And we found that our wild type LPS is really abundant in galactose, which is the same component of the galactan. When we looked across our PAM mutants, we noticed that all of our PAM gene mutants underwent a pretty considerable decrease in galactose. Again, supporting our hypothesis that this HMW LPS, this control strain LPS, is really abundant in galactose, which potentially may be from galactan. So in order to confirm that the source of galactose is this galactan, we ran out purified LPS samples on a gel and used our GP19 antiserum to do a Western blot. So what you'll see here on the left is the Western blot, and you'll notice that we observe reactivity in our control strain LPS, and then this is the surface wash just as a control, but that reactivity is lost in all of our PAM mutants. Here on the right, I'm showing you the same data, but in a bit more of a quantitative fashion. This is an ELISA, and the plates have been coated with purified LPS. And again, you'll notice that we see this really high level of reactivity when we coat our plates with wild type or control strain LPS. And this suggested that the galactose that we're seeing in our LPS is coming from galactan specifically. And furthermore, that you need a full PAM locus to be expressed in order to detect that reactivity, in order to get that production of that HMW LPS. So we think that you need PAM A, B, and C to produce the galactan, and then expression of PAM D and E in order to anchor that galactan to the bacterial surface, presumably via a connection to that atypical O antigen. So just for a little visual representation of what I mean, we think in our HMW LPS, we have LPS molecules that have lipid A and core, the PAM D and E dependent atypical O antigen, and then the PAM A, B, and C dependent galactan that is potentially covalently attached. And then in our LMW LPS, we have molecules that are composed of lipid A and core, and then the PAM D and E dependent O antigen, with every band in the ladder representing another repeating sugar unit in the O antigen. So in order to prove that these LPS configurations are what we think they are, ideally what we wanted to do is identify this specific linkage using NMR. So to attempt to do this, we again worked in collaboration with the CCRC, and we separated our HMW and LMW LPS clusters by size exclusion chromatography. And so you'll see we have these really nice clean fractions. Fraction 2 is our HMW LPS, and fraction 3 is our LMW LPS. And then we did an NMR analysis to identify the linkages present. And so I want to direct your attention to the peaks in the box here. So we found that in our fraction 2, our HMW fraction, excuse me, we have galactofuranose residues of the same specific linkage that has been previously shown to be part of the galactan exopolysaccharide. And so that confirms that our HMW LPS contains galactan. And now if you sort of look at this other portion of this spectra, we have all these LMW LPS fractions that are really, really complex. There's a lot of peaks, there's a lot of noise. And we expect that that is due to the presence of all of these other sugar residues in the core and in this atypical O antigen. And this complexity combined with what we expect is a really fragile, very single linkage between our two components those two caveats kind of prevented us from using this analysis to identify that one specific linkage that we're looking for. And so to circumvent that, we took a different approach. We decided to look for lipid A in each of our LPS cluster or LPS fractions. So we hypothesized that because lipid A is the initial component of LPS upon which all subsequent residues are built, that if our atypical O antigen and our galactan are truly attached to each other, then both of these fractions should contain Kingella-Kingay specific lipid A. So what I'm showing you here are the MALDI-TOF spectra from each of the fractions. And what you'll notice is that the spectra match. So we identified the same major ion species in each of our HMW and LMW fractions. These are the structures that correspond to these major ion species. They differ just in the presence of some of these phosphate and phosphethanolamine decorations, but the base ion peak is the same for all of these. And in data that I don't have time to show you today, we confirmed that this is Kingella-Kingay specific lipid A. It is a unique lipid A signature. So we know that this isn't coming from somewhere else. And so overall, this provided us with some really strong evidence that our atypical O antigen and our galactan are attached, and that results in the production of this HMW LPS species. So I want to wrap it up just by summarizing some of our main points with our working model of the Kingella-Kingay outer membrane. So we think that in our control strain, we have this heterogeneous population of LPS molecules where we have this LMW LPS that's composed of lipid A, the core oligosaccharide, the PAM-DNA dependent atypical O antigen, and then we have our HMW LPS molecules, which are composed of lipid A and core, the PAM-DNA dependent O antigen, and then the PAM-A, B, and C dependent galactan, which we believe is covalently attached. In our PAM-A, B, and C mutants, we have just a pool of LMW LPS molecules. And then in our PAM-DNA mutants, we observe just a truncated form of LPS. And we note that in the absence of PAM-DNA, the galactan is absent from the surface, although there still remains a question as to where it goes. But that data really implies that PAM-DNA are really critical for galactan surface anchoring via production of this atypical O antigen. So overall, this is a really exciting phenotype for us. It represents a new mechanism of exopolysaccharide anchoring. And we hope that this model will inform our future compositional and functional studies and how we and really help us understand how these polysaccharides work in the context of infection, given this sort of novel interaction between an LPS and a bacterial exopolysaccharide. So there are several future directions. Just immediately, we're really interested in understanding the individual roles of the PAM genes. We have some guesses, we have some data here and there, but we really want to do a more rigorous analysis of these gene functions. We're also really interested in understanding where in the pathway these two, or where in the cell and where in time these two biosynthetic pathways collude. What space are these polysaccharides assembled in, and then when do they cross paths to exit the cell and get to the surface? So lots of fun stuff to do, and we're really excited to continue this project. So with that, I just want to thank my lab, my incredible collaborators for their help, especially with all of the NMR, my thesis committee, my funding, and of course you all. And then with that, I will take your questions.
Great job, Nina. That was a really, really great talk. And yeah, so if anyone has any questions, please feel free to pop them in the chat, and then I can relay them. But to get started, I have a question, maybe sort of two questions. I guess, do you think maybe that these two high molecular and low molecular weight LPSs might be differentially immunogenic? And sort of along those lines, do you think that there are different conditions, potentially like stress conditions for the bacteria, that might lead to preferential composition of one versus the other on the cell?
Those are such great questions, both of them. So with regards to the immunogenicity, so we have started to look at how these different LPSs activate TLR4. So TLR4 activation is primarily driven by the composition of lipid A. And we do know that our lipid A is hexaacylated, which lends itself to being pretty inflammatory. But one of the things we've always been interested in is how these really large, seemingly very large polysaccharide components change LPS micellar geometry, which is sort of what determines how the how easy it is to get an LPS molecule to the TLR4 MD2 pocket. So we've done a few of those stimulation assays, and so far we don't really know. It's not super conclusive as to whether or not this really large LPS kind of changes the way that TLR4 gets to interact with our lipid A, because it seems to be pretty inflammatory across the board. We do have some tweaking to do with the assay. We also have to, we're trying to make sure that we're not saturating our system and getting just an overwhelming amount of LPS that's out of my cells, because it's so hydrophilic given this really large polysaccharide component. And so we're trying to best tweak the assay in order to get at some of those questions, but that's a really, really great question. In terms of stress conditions, we haven't really done a lot in terms of tweaking environmental stress conditions to see if that changes the presence or the ratio of LPS species on the cell. We don't actually know what the stoichiometry is under, like steady state conditions, but would love to know, because certainly there's precedent in the literature for LPS switching, based on environmental stress. We know that sometimes in OMVs, LPS can be preferentially switched to a different composition on OMVs. So lots of great questions there as well. Wish I had a better answer. Wish we knew.
No, that's great. Okay, and then Dieter asks, isn't it surprising that the high molecular weight LPS is one nice band on the LPS gel?
Yes.
This suggests a defined number of galactin added to the LPS.
Yes, great observation, and such a great question. I think about this a lot, actually. We do find that it's really interesting that we have, this seemingly very narrow distribution of sizes. Typically, in something with an O antigen, you get a really long ladder. And what that implies to us is that potentially the galactan is very limited in the amount of sizes that it can exist in, how many residues get added. And what that also suggests is that there's a very tight mechanism of chain length regulation, which is what sort of determines how many residues get put onto each of these various components. We also don't know if we have more than one mechanism of chain length regulation going on, since we have two different polysaccharides that are attached to each other. Would love to know. But yeah, it is very surprising that we have one really clean band and then what seems to be a very narrow ladder, and that they differ quite significantly in size, because it does suggest that there's some sort of threshold or some critical residue that has to be added, and that, immediately results in galactan addition all the time. So very, very interesting.
Awesome. Well, thanks so much, Nina. Great job again. Super, super interesting stuff as always. And so, yeah, I think next up on our schedule, we're scheduled to have a short break, although we are running a touch behind on time. Matt and Kayla, I don't know if we maybe want to modify the length of the break slightly. What do you guys think?
Yep. So I think we'll switch it to a 5-minute break, give everybody a chance to take a little break and then come back in 5 minutes, and we'll begin from there again. Thank you.
Sounds great.
Okay, it's 5:20 in a few more seconds. Okay. We should be coming back soon. It's 5:21, and we can get started with the second half. Dr. Kutzler?
Yes, thank you so much. Thanks, Kayla, and thank you, Matt, and to all the student officers. I just want to take one moment to make an announcement to our branch, a very important and very integral member of our branch. Dr. Jim Poupard was a longtime member. Just really quickly, for those of you who may not know who I am, my name is Michelle Kutzler. I serve as the president of the Eastern branch of ASM, Eastern PA branch here. And I just want to take a moment before our next speaker to announce the passing. Dr. Jim Poupard was a 50-year member of National ASM, and as well as our branch. He was just an amazing individual. Many of us know him. And I just want to take a few minutes to talk about Jim. He was, for those of you who don't know, he was born and fully educated and lived all his life in Philadelphia. He went to Temple for undergraduate, he went to Jefferson for his master's in clinical microbiology, and he went to Penn for his PhD in interdisciplinary sciences. And since 2003, he was president of the Pharma Institute of Philadelphia, which was a consortium of consulting companies that specialized in antibacterial drug discovery. After obtaining his PhD at the University of Pennsylvania, he was the supervisor of the clinical microbiology lab at the Hospital of the University of Pennsylvania. He was also the microbiology director at Bryn Mawr Hospital and associate professor of pathology and medicine at Medical College of Pennsylvania. So you can see just from that short intro how many institutions in Philadelphia he was just a part of their fabric. So truly an icon in Philadelphia. In 1990, he was named director of clinical microbiology at SmithKline. And then after some time, he returned to work at the same company, later named GSK. And so with over 100 scientific publications, Jim was recognized as an expert in the area of anti-infectives, as well as science policy, and what I'm going to spend a little bit of talking about, history of microbiology. He became, as I mentioned, a member of National ASM in 1971, and he twice served at National as the director of the Committee for the History of American Microbiology. So he was instrumental in creating and serving as chair of the Center for History of Microbiology, the ASM Archives, located outside Baltimore at the University of Maryland, Baltimore County. And for our branch, he served as the chair of the EPAASM branch archive, and he contributed so much to the history content on our website. And he's truly what we call our historian in chief. So he's going to be greatly missed. For those of you who don't know, the book that I'm showing on my slide here, he authored, and it's called A History of Microbiology in Philadelphia from 1880 to 2010. It's just a wonderful history that, I have this book sitting on my shelf in my office. And, this book takes readers back to the 1880s, where bacteriology started to become identifiable discipline of science, and it began to be separated from established fields such as pathology, as well as histology and microscopy. And so the first part of this book really focuses on the people of Philadelphia that played an important role in microbiology, bacteriology, and also the institutions that really helped Philadelphia shape to become a great city for the history of microbiology. And he also talks a lot about how bacteriology became to be more aligned with medical school curriculums. He really summarizes an entire generation of bacteriologists who contributed to the formation of, across all of our institutions in Philadelphia, academic departments of medical schools, research institutes, pharmaceutical companies. He really bridged so many institutions. So This book really beautifully summarizes the evolution as it progressed in Philadelphia, with an emphasis on the role of our branch in particular, and the importance of this organization really establishing Philadelphia as the center for teaching and research. And I just am just so saddened by the news of his passing, and I wanted to share it with everyone today. And we will be certain as a branch to make sure that his legacy continues, and other activities and other events that we're planning. I just want to mention a few other things, and importantly, even in towards the end of his life, when he wasn't feeling well, he still was continuing to write. And he recently authored Ruth Emma Miller, PhD, who was our first female president of the Eastern PA branch of ASM, and all of the female president's successors. And that's housed on our history page. So even towards the end of his life, he was still authoring important works celebrating our branch. So I just want to end with a few comments and thoughts from some of the members of the executive committee, including Linda Miller, who served as the co-director of the archives, and she was a longtime colleague of Jim. She's on the call today. If you don't mind, Linda, I'm just going to share some of your thoughts. So she said, for those of you who knew Jim, I know you understand how much our branch has lost. His love and enthusiasm for the history of microbiology was contagious. His insatiable passion for clinical microbiology, the history of science, his love of life were a treasure and a gift to those of us who knew him in this world. And so I just want to thank everyone for allowing me to take a moment to celebrate Jim. He really was a dear friend and colleague to many of us at EPAASM. He was a wonderful colleague and mentor, a leader, a driving force, and really dedicated supporter of our branch for many decades. So with that, this May student branch hosted monthly meeting was one of Jim's favorite events. He enjoyed listening to the student talks. He often said how trainees should understand what lessons Pasteur could teach them about their dissertations. And he also really felt that our branch members should understand the significance of the magnificent branch history, and how all of us that are working in the field of microbiology really are contributing in Philadelphia, making us really an important part of the history. So I'm going to next turn this meeting back to our student officers to introduce our next speaker, Dr. Josh Hamel. And I know Josh is also going to use a portion of his talk to really talk about his own career path in microbiology. And I think Jim would be very pleased to listen today and to learn from all these next generation microbiologists. So thank you very much for a few moments that I could just talk about Jim, and I turn it back to you.
So I'll be doing the introductions. Thank you so much, Dr. Kutzler, for the great memorial. Let me just share my screen. Let's see. Okay. Good afternoon, everyone, once again. Today, I have the great joy of introducing our keynote speaker, Dr. Josh Hamel. Dr. Hamel did his dissertation at the University of California, Davis on Escherichia before doing a postdoc at the University of British Columbia. There he began his research on H. influenza, and now he has brought all of his expertise and work experience and life experience to become a valued member as a professor over at Drexel University College of Medicine. While doing all of this, he was publishing numerous papers along the way, and Dr. Hamel has pushed our understanding of genomics. He is known, especially amongst the students, for his kindness and is always willing to learn new subjects as well as drive science as it should be driven. And I believe I can speak for all the graduate students that by saying that Dr. Hamel is a blessing and has taught us so much. So before I can blather too much and waffle on, I just want to turn it over to Dr. Hamel and welcoming him and just saying that he will be doing his talk on the mechanisms of genetic change in the natural history of the pathogenesis of H. influenza. Dr. Hamel.
Thank you so much, Kayla. That's very nice of you to say. Sometimes I think that maybe I get a little distracted from things I should actually be doing, but I can't really say no to students very easily because I remember still a little bit being one of you. So let's see if I can share the screen here. Everything good? All right, so I don't really know exactly how to work in the travelogue kind of scientific journey part, but I will. I'll try to hit a couple transition points at some point along the line. But my real interest over time has been genetics. I got a PhD in genetics, and what I learned as I went along is that I should also learn about genomics. So I do a lot of that now too. And I wouldn't have gotten anywhere without a huge number of people. The work I'm going to talk about today, pretty much all of these people have had some role in the work I'll talk about today. I should highlight some mentors, my own PhD advisor, Sean Burgess, and my postdoc advisor, Rosemary Redfield, both women in science, both who ran very small labs and had to kind of confront the difficulties of being a woman in science during their sort of upbringing at their age, and taught me a lot. I also should thank my senior faculty mentor, Garth Ehrlich, who has really helped recruit me to Drexel and helped me find myself a faculty job and so on. I also have some wonderful collaborators that I met at a meeting in my postdoc that I still work with very closely that I should mention, Juncal Garmendia, who's in Pamplona, Spain, and Dr. Paul Planet, who's over at CHOP. I have published multiple papers with Juncal and have multiple grants with Paul. And it's funny how you just meet somebody at a conference one day and you stick with them for the next 10 years. And I should also thank all three of my PhD students that I've ever had have all recently graduated during the pandemic. I'm very glad that they've all kind of moved on to the next great steps, but they've also been instrumental to my development. And also my long-term senior technician, Lynn Hammond, and my long-term senior bioinformatics programmer, Rachel Ehrlich. Okay, so I'm going to just start with a quick introduction about where I'm going, and by that I mean not only this talk, but like me, because I didn't really come into this knowing anything about bacteria. I started learning about bacteria when I was about 30. I used them for cloning before that, but that was really kind of it. And so where I will be going is to talk about the fact that so many of the things that I thought were bacterial pathogens aren't, at least not as a part of their professional lifestyle. They only incidentally cause disease. They're normal commensal residents of our bodies, and it's almost just a pure accident that causes them to end up causing disease. It's not clear in a lot of cases for many bacteria that they really gain any benefit at all from the diseases that they cause. Because of my model organism that I've been studying since well before I worried about these problems, is the nasopharynx. I don't have to introduce that location of the body anymore. But I think I should mention that it's just a very transient, strange environment to specialize in where you ought to live, right? As a niche, it's odd. It makes sense because these bacteria transmit through snotty-nosed kids a lot of the time, but it also allows them to fall down into the lower respiratory tract, to get into the middle ear, cause sinusitis, and even, in other cases, cause invasive diseases. Because they're not really trying to cause pathogenesis, it's kind of hard to understand what a pathogenesis factor is. We can get into that a little bit, but basically I should just mention that the species that I'm going to be talking about, of course, is Haemophilus influenzae, which is one of these highly prevalent bacterial organisms that we might call an opportunistic pathogen, but I don't know what opportunity Haemophilus gets from causing meningitis. Or they're commensals that can sometimes have pathogenic behavior. There's a lot of factors outside of the bacteria themselves that lead to them causing these diseases. Probably one of the most prominent ones is respiratory viral infections, particularly influenza virus and RSV. Also, a lot of host factors related to the immune system of the host, delayed immune development in children or compromised immune development in others. Predisposing conditions, again, former smokers, people with chronic lung conditions, and various other risks. Because it's such a strange niche living in the nasopharynx, there's a whole lot of things that these bacteria express that might be called virulence factors, but they probably also need these just to be commensals, just to live their normal life. They got to be able to live in the mucus. They've got to be able to stick to the airway epithelial cells. They've got to be able to evade the immune system through any number of complicated mechanisms. They often form biofilms, sometimes together. And whether or not these various factors lead to disease is multifactorial. But it's worth noting that these 3 species that I'm talking about here all have many of these types of mechanisms. I'm going to specifically hone in on Haemophilus influenzae, which is a host-restricted organism. It only lives closely associated with like one host. So Haemophilus influenzae is only found in nature in humans. And most of the other species in the family to which it belongs, the Pastoralaceae, are associated with one animal. So like A. pleuropneumonia lives on pigs, Mannheimia hemolytica lives on cows, and so on. It's a naturally competent bacterium. I'm going to spend probably half the talk on that. We'll see how it goes. I got a lot of slides. You asked for a scientific journey, so we'll go on one. Natural competence is a state where bacteria actively transport DNA from outside to inside, where it has the opportunity to recombine with the chromosome and change the bacterium's genotype. And just like many other species, Haemophilus influenzae is incredibly diverse. The level of diversity that distinguishes 2 random isolates of this organism is bigger than the diversity between most primates. So if you were to pick up the first cellular genome ever sequenced, which is from Haemophilus influenzae RD strain, and you compare it to the second Haemophilus ever sequenced, then you find an enormous amount of genetic difference. You find a SNP every 15 base pairs instead of every kilobase and a half like you'd find in us. You find hundreds and hundreds of structural variants, insertions and deletions and duplications and inversions and transpositions that distinguish these 2 strains. You can take it to a bigger, broader class of strains. So this is, I'll talk a little bit more about this data set later, but this is showing a gene possession heat map of where the columns represent individual strains' genomes and the rows indicate individual genes. What you can see is that all of these strains share a core genome of over 1,000 genes, but they actually have twice that many genes that they don't share across all strains. And so that level of difference is just wildly different than anything that we would encounter in a eukaryote. And we'll get to a little bit of why that is in a second. So these are highly divergent, not only in their gene content, but also in the divergence among the alleles of the genes they contain. This is another way of showing sort of the same type of information, but without a scale. At Drexel, we've sequenced over 1,000 strains of clinical isolates of Haemophilus influenzae in combination with many collaborators, and we've combined them with resources at the NCBI to build sort of a huge phylogenetic tree of all of the diversity of the species and from a variety of disease contexts, and we work with this data set quite a lot. You can see one of the things that's often difficult with an organism like this, and this has changed a lot, thankfully, I think, to a lot of microbiologists, is if you were to look at the databases 10 years ago, almost every single isolate you would find was from somebody with disease. And given that they're far more prevalent in people that don't have disease, it's good that now we have a lot of strains that come from healthy children. All that genotypic diversity, genomic diversity, correlates with a lot of diversity among strains in their phenotypes, as you would expect. Pretty much anything you care to measure. The type B capsule of Haemophilus influenzae used to kill about 500,000 children a year. It was this one clade right here that's all been vaccinated away and no longer really much of a problem. But any other kind of trait you care to measure has quantitative or qualitative differences. And there's a lot of ways that these organisms can change relatively rapidly. It's deep divergence. It's like looking at something like primates or something like that. But in fact, besides just long-term divergence through asexual reproduction and mutation, there's also some rapid mechanisms. I'll talk a little bit about natural transformation, but I would also be remiss to not talk about phase variation, because this is going to come up a couple of times. And this is a property that about a dozen-ish genes in a Haemophilus influenzae genome have, where there are simple sequence repeats within open reading frames, and the multiple of the simple sequence repeat is not multiple of 3. So when DNA replication makes mistakes, that kicks proteins in and out of frame, so they turn on and off, and that can happen at a much higher rate than normal mutations. This has sort of been, I would say, a positive control. I'll call it that in retrospect. So where I've kind of come to in my sort of, I'll call it the end of my early professordom, is to sort of ask these big major kind of questions using the diversity of bacteria, this one species, as a lens to organize my thoughts around health or disease and how this diversity impacts these transitions between commensal and pathogen. Can we isolate natural bacterial variation associated with pathogenesis? I'll talk about this both in an experimental context and also in a sort of more statistical context. And at the bottom of that, then, what are the molecular mechanisms that distinguish commensal and pathogenic strains? Is it all just host and environment, or are there things that we can look at in the bacteria to help distinguish the ones that will cause illness in a child versus the ones that won't? So that's where I've kind of gotten to and what my major kind of important questions are to me now. But just to give some backdrop, I don't really know how other people do this, but I'll just do it this way. I'll give you a little quick biography, and then I'll talk about some major transitions and some other ways I did that. So, being an Eastern Pennsylvania branch of the ASM is interesting for me because I'm from the West Coast. Kayla asked earlier if I was excited for the summer, and I absolutely am not. I grew up where there was only spring and fall, and I've come to a place that only has winter and summer. So I don't know how that happened, but here I am. Went to college in the Bay Area. After college, I worked in some biotech companies. That was a really great experience. I loved it, actually, and I was sort of driven back to school by the glass ceiling that I had there as a bachelor's of science. And then once I went back to academia, I never wanted to leave again. Going back to academia as a tech was a 50% pay cut, and going from a spaceship to a garage. It was like completely different, but people cared so much about what they were doing, even if it was the evolution of cricket songs, that it really kind of compared to what the environment was like in industry. I just preferred that for my life. I wonder if that was the right decision now, but here I am, and I can't imagine going back at this point. I did sort of start out as a microbiologist, but the kind of microbiologist that pretends they study humans. So if you study Saccharomyces cerevisiae, you try to get your funding from cancer research dollars and like birth defects related research because of the way that budding yeast is used as a model. There I studied meiosis. The link with meiosis will become somewhat clear to the rest of the talk in a minute. And afterwards, I actually kind of went on an itinerant wandering postdoc because I wanted to learn about genomics, learn how to use computers, and I wanted to go back to thinking about evolution in my future career. And I needed to go bother some friends and help them start their labs while they taught me how to use a computer. Then I did a proper postdoc at the University of British Columbia on the West Coast, where I've still spent more than half my life before I landed at Drexel University in the Center for Genomic Sciences. So I'll get through sort of what I was doing in my postdoc and why I was doing it, and then how my questions and thought process changed as I looked for and obtained a faculty position. I welcome questions at any time. For the students or anyone else, you can ask me questions about the science or the rest of it whenever you feel like it. So kind of just backing up, all living things that have genomes and replicate have, as the ultimate source of all their genetic variation, is mutations. There's a lot of different kinds of mutations. They're not just point mutations, but in the end, it all comes from that. The problem is when you acquire a new mutation and you're only clonally reproducing, those mutations are stuck in the lineage they arose within. If this cell got an orange membrane, this other cell that had a yellow cytoplasm can't get an orange membrane unless it gets the same mutation in the same gene and the same allele or whatnot. So that causes all sorts of problems in evolution, and people have talked about this for hundreds of years at this point. One example that we talk about a lot in bacteria is clonal interference, where if you imagined that both of these mutations led to benefits to the organism, the problem is that while they both might go up in frequency, eventually they'll start competing, and you can't get both of them into the same lineage until you wait for that double mutant to arise over time. And there's a whole bunch of other related problems that evolutionary biologists have used to try to explain the existence of sex and non-clonal reproduction. These problems are all worse in small populations, and I don't even know how to define a population of bacteria genetically, really, because that's still a very complicated question. But the problem is worse the fewer individuals that there are in a population. So in textbooks and the way we're taught in biology, that the solution to this that eukaryotes have arrived at is mating and meiosis. So two haploid cells fuse and mate to become a diploid cell, and that may grow into some organism or it may not. It may immediately undergo a reductive division that yields a new diploid cells or new haploid cells that are recombinant. This happens across all known eukaryotic organisms in a very kind of sporadic way. It's often facultative. It's only rarely obligate. It's obligate in most animals, but even it looks like a lot of animals have tried to evolve away from it if they could, but they're stuck with it for various reasons. One of the things about this process of mating and meiosis is that it's very, very evolved. The meiotic process is this incredibly intricate orchestrated dance between these two genomes where they reciprocally exchange material, and it involves the entire genome. And this is what I did for my PhD. So I studied it in the budding yeast. So I pretended I was studying human, and I focused on the very early steps of meiosis, of how the homologous chromosomes paired up along their lengths during the early stages. And so even though I was really drilled into the mechanism a lot, I did like talk to a lot of evolutionary biologists and wonder about why this process existed. And just as a segue, when I got to the end of my budding yeast PhD in genetics, I was trying to figure out what to do next. And I probably was going to get out of meiosis, but I thought I was going to go work on like a higher organism, like a fly maybe, or a nematode, or maybe a mouse. I didn't really know, maybe chromosome biology, maybe chromatin. I had to cast a very broad net. I was also hunting simultaneously with my partner and now spouse. And I asked a lot of people, what do I do next, right? And they all said, not yeast, get out of yeast. Mouse is the new yeast. And that was a while ago. So just to kind of give the context here is one of the main reasons budding yeast became such a powerful model system is because you could target mutations. And as soon as that started to develop in higher eukaryotes, and especially in mice, the utility of having the budding yeast kind of went down a lot. And a lot of my advisors who worked in yeast were worried about that, and they were actively trying to develop new model systems in order to deal with the fact that their funding was going to start drying up, because you can't study the cell cycle in yeast to emulate cancer if you can produce the same cancer and emulate it in a mouse. But more kind of broadly, the really good piece of advice I got that somebody phrased really nicely was to shift, but not necessarily what I do with my hands. So do something where you're going to land, hit the ground running quickly so you can start doing the work, but make sure that you've completely shifted what you read so that the literature you read and absorb is not the same anymore. So you grow your brain, you do something different. And that I really like that advice. And landing in a bacteria lab, that was real nice because I could hit the ground running quite easily. I had already spent the last half decade or more of my life counting colonies and doing serial dilutions. So that all kind of came naturally. I had to streak a lot softer, right? But most of the techniques were really quite similar. So the transition here is the similarity is that bacterial natural competence is the one process in bacteria that resembles sex. There are other processes that move genes around in bacteria, but they do not resemble sex. Conjugation of plasmids and transduction by viruses, those are parasitic processes, right? We don't call the insertion of a HIV into a genome, we don't call that sex. We call that a proviral insertion, right? So the reason it's so different is that the bacteria, in the case of naturally competent bacteria, they are actively taking up the DNA. They're doing the work to bring the DNA into the cell. It's not because some donor is forcing this DNA into them. So that's the really fundamental distinction between this process and the others that move genes around. And the really fundamental distinction between eukaryotic sexual reproduction, there's many, but from a genetics perspective, really the fundamental one is that it's non-reciprocal, it's directional, it goes from a donor to a recipient, and it doesn't involve whole genomes, it just involves segments of genomes. It involves the pieces of donor DNA that the recipient cell took up. So why does this happen? I mean, the model is sort of what I showed before. The idea is that we can break free from this restriction of mutations to single lineages. We can purge deleterious mutations by recombination, or we can add together beneficial mutations by recombination. And so we can accelerate the pace of evolution. And that's kind of the classical explanation for the evolution of sex. And it's one of the main explanations that's given for why bacterial natural competence exists. I should be mindful of the time because I think I have vastly too many slides. So I'll have to go speed it up a little at some point. Is this a sufficient explanation for this trait, sex or competence? Probably not. I don't have to dwell on it too much, but in eukaryotic organisms, that's actually pretty rare. They do it quite often, but just enough maybe. There's problems besides recombination that meiosis and mating might solve. And in bacteria, it's even more strange. Like, why are they taking up their dead relative's DNA and recombining it into their chromosome? It's weird. Like that's a strange thing to imagine it being beneficial. So there are alternative explanations, and one of them that my postdoc advisor, Rosie Redfield, championed for many years is that it's for food. And that's a pretty good explanation, and that the recombination is actually incidental, or as a side effect of other processes. And I'll bring up quite importantly that if you delete the machinery that is required for natural competence, you also lose some adhesion organelles, the ability to move around, and the ability to form biofilms. So it's quite possible that this machinery is just pleiotropic, it does multiple things. Why is this important? Well, it's all over the bacterial phylogenetic tree sporadically, and it's a medical threat. This is how bacteria can share each other's genes. So for naturally competent bacteria, that means you can rapidly become antibiotic resistant. If you have a nearby relative with antibiotic resistance, you can toggle your cell surface antigens, you can acquire or delete virulence factors, and so on. So I haven't shown this in a long time, and it's getting a little dated, but my postdoc advisor, Rosie Redfield, recently retired, and just in honor of that, I'm going to do a quick illustration of the mechanism of natural competence as seen by Rosie Redfield in her stop motion animation video here. So these organisms become naturally competent when they are starved of their preferred sugar and turn on their CRP response, and when they are starved of the precursors to purines. When that happens, the starvation of PTS sugars induces the transcription of the sexy transcription factor, whose translation upon depletion of purine precursors modulates CRP's binding to turn on this 26 gene operon or regulon, several different operons, sorry. These components include an outer membrane type 2 secretin pore, an inner membrane, highly conserved inner membrane motor pore, some accessory factors that help restrain DNA that's been taken up into the periplasm, and this type 4 pilus motor complex, and type 4 pillins. So DNA is not something that can just walk into a cell. It's stiff, it's charged. It can't get through these membranes without active work and active deformation of the structure. The energy of ATP is used to power the assembly of a type 4 pseudopilus. In Haemophilus, it doesn't really extend much beyond the surface of the cell. And there's a sequence motif in the Haemophilus influenza genome that's highly enriched, that specifically interacts with the tip of that pseudopilus, causing some type of switch in the ATPase activity that disassembles the pseudopilus and pulls DNA, kinking it through this pore. This next part has been proven wrong, this by Melanie Blokesh and Vibrio. This protein doesn't actually restrain the DNA to allow for multiple cycles of pilus elongation and retraction. Instead, it just polymerizes along the DNA and helps ratchet the DNA into the cell. And that's because these bacteria can take up very, very large pieces of DNA. We still don't know the limit on how big that is, but many times the length of the cell. So they can't just initiate this loop, they have to do something else. The inner membrane pore takes a free end and it degrades the 5' strand while inserting the 3' strand into the membrane. If that DNA has no homology to the chromosome, or even if it does, most of it's degraded. But if that DNA comes from a close relative, then REC-A-mediated recombination can add it to the chromosome. And that's how the mechanism works. Just to note, as a quick note in terms of the function of natural competence, none of the recombination machinery is induced. It's all, how do I switch back to the other one? Excuse me, guys, sorry. It's all there because it's all part of the normal DNA repair machinery of the cell. So Haemophilus has been used since the 60s as a model for natural competence. There's other models. Pneumococcus was the first. That's how we discovered DNA was the genetic material in the first place, was doing a transformation experiment. And so we know a lot about it. I'm just going to reiterate 2 points. One is there's this weird sequence that's almost every single kilobase in the genome that massively increases the probability that the DNA can be taken up. It's sufficient and necessary. It's unclear what it's there for, but it certainly is highly overrepresented. And it certainly means that Haemophilus influenza vastly prefers its own DNA over that of other organisms. The other point is that divergence between two DNA molecules reduces the rate at which they recombine. If we feed, so there's a typical experiment. These are quite straightforward. You take the Haemophilus influenza exponential culture, you starve it in a starvation medium for a couple hours, and then you can add marked genomic DNA and get really quite high transformation rates in about 15 or 20 minutes. If you feed DNA from a derivative of the cell you're using as a recipient, the rates are better than if you use a diverged clinical isolates DNA. So for my postdoc, I really wanted to kind of address these first two questions. And then the transition into my faculty position, I wanted to start thinking about this third. So people have been doing these experiments from the 60s, but because we didn't have like these super fancy DNA sequencers that were really cheap to use, we didn't really actually know how much cells took up and recombined DNA into their chromosomes. We could sort of estimate it, but there was no real direct way of doing it. There's some evidence from various sources that suggests that there could be hot spots and cold spots of recombination, but it's also not entirely clear whether that was true or been tested in a really rigorous way. And then I'll transition into this question about how we could influence, how it might be important or not a little later. I'm going to go quick for a little while because I want to make sure I get to the more current stuff. And this has been published for a while. But the premise here is pretty simple. This is a 1965 experiment with a DNA sequencer. One twist is that rather than using antibiotic-resistant derivatives of the lab strain, we're using these clinical isolates that carry tons and tons of genetic variation. So that if we transform the cells and pick up clones and sequence their genomes, we can identify where the recombination tracks are because of all those markers distinguishing those two strains. This was done a while ago, 10 years ago, oh my God. And this was the first sequenced transformant. We selected for somebody that picked up an antibiotic resistance marker, and you can see that it picked up that and a lot more, plus some unselected tract. These recombination tracks span regions of low and high nucleotide divergence. You can sort of see by the density of lines. Each of these lollipops is a SNP that went from recipient to donor. This axis is the genome length, the recipient genome coordinate, and the frequency that we see the donor specific allele at that site. This scale of this is dozens and dozens of genes in one transformant. It's not just changing the alleles of those genes. Sometimes it's adding or subtracting genes. There's disruptions in these recombination tracks that probably correspond to sites of some kind of mismatch repair at sites of structural variation. And we can do this for a bunch of clones. We can look across many selected transformants at one site and another site. And what I think you can all appreciate is that we got recombination at a lot of places other than where we selected for. There's a lot of unselected transformation. And in these experiments, if you look around the selected site, you can sort of see a decay away from where we selected of donor-specific allele frequencies corresponding to the DNA fragments that specifically recombined into each of those clones. In these experiments, on average, about a % of the genome was replaced in multiple different independent segments. We couldn't really see any obvious effective divergence on transformation. We did see a weak bias of deleting genes over inserting genes, which is kind of interesting. There's also sort of an interesting point to be made here that across this set of just 72 clones, we had about a third of all the donor-specific single nucleotide variation present, sampled from the donor genome. So this both sort of indicates how easily this DNA can move into these other cells, but from both like a natural environment perspective, but also from the perspective of maybe we could map genes with this. And so I'll get there in a second. But the other observation that was noted by my graduate student, Danielle, is that the recombination tract length, the longest ones that we saw, these longest recombination tracts, they were just about the size of the genomic DNA that we were putting in the first place. So this might have just been restricted by the fact that we were purifying genomic DNA on average about 50 kb the way we were doing it. And so that was the limit. So maybe there's actually another limit. And so she set out to see if that was true. And she actually did old school agarose plug preps of DNA. So she could actually get a distribution where some of the DNA was real long. And these preps on average, the average length was about 100 kb, and maybe about 15% of the DNAs were more than 250 kb. And we had this strain that would allow us to check whether co-transformation of really distant markers became possible under these conditions. So the way this works is we have a nova bias and resistance marker. It's about 15 kb away from a streptomycin marker. It's about 250 kb away from a spec marker. That's more than 10% the length of the chromosome, right? And then a really far away one where we would rarely expect them to co-transform. So this is the genetics. So the genetics says if we have two unlinked markers that are on opposite ends of the chromosome, doesn't matter what size of donor DNA we see, the observed to expected ratio of double transformants is around 10. The reason that we see more observed than expected is because only a proportion of cells in the culture are competent. And so we see this excess. So this is sort of the control of unlinked markers. If we have tightly linked markers, it also doesn't matter whether we use medium or long DNA. We see a much elevated observed to expected ratio because the vast majority of molecules in the preps span that whole interval. And then finally, when we use these sort of intermediate, about 10 or 15% of the chromosome length apart, we do suddenly see a difference where our medium length fragments fail to show elevated co-transformation above the control, but when we use the very large fragments, they do. So this suggested to us that we're not really at a meiosis level, but it is possible for these recombination tracks to be much longer than we had thought. And this might actually have some real in vivo relevance because of the way that the bacteria may interact in nature. It's really dependent on how quickly the bacteria is taking up DNA after its relative died, and how much the shear forces and various other things digest that DNA before it's taken up. So just to quickly look at this, another experiment that Danielle did was to take these really long DNAs from diverged clinical strains that she'd added markers to, and pool thousands of them together and then sequence them to high coverage and look at that decay in transformation away from the selected site. And part of that was to confirm that do we indeed see these really evidence of really long distance co-transformation, and then also what would be the effect of rearrangements. So just to show you a quick example from that work, here's three genomes being compared by whole genome alignment, and these colored blocks represent locally collinear blocks, and you can see there's regions of missing and rearranged DNA among them. She made a bunch of different marked strains. She did a bunch of pooling and sequencing of transformants, and I'll just show you this quick example of this region around this spectinomycin marker, resistance marker, where this pink block of genes, about 25 genes, has relocated between the recipient and the donor. This is what the results look like. That's on a log scale. Right where we selected, 100% of the strains acquired the resistance marker. And then as you march away, you see this kind of monotonic log linear decline in the transformation frequency, except at some places, right? So here, there's this big declines around this phage that's in the recipient, but not in the donor. There's also this weird decline here, followed by a really long distance, 150 kb gap, followed by more transformation. But this makes perfect sense when you instead show it on the donor coordinates. It's because the donor DNA that was being taken up, of course, was more likely to have that pink block nearby. There's a decline right around where the rearrangement breakpoints are, but there's really no barrier to transformation beyond those local breakpoints. That's an important point about the difference between transformation and meiosis, is that meiosis enforces synteny. Organisms that undergo meiosis with each other, the gametes will die and fail to form properly where crossovers happen within these rearrangements. In bacteria, there's no such barrier. So bacterial transformation can have these long distance effects, even if it looked like a long distance effect, even if in reality it's quite close together. Okay. So this next bit is also unpublished, and coming from Danielle's work and some of my early old work. And this was to ask the question of whether some parts of the chromosome transform better than others. And the idea here was quite similar, but rather than taking this huge high molecular weight DNA, we can shear it down to a much smaller size and then transform cells and then pool many recombinants and then do very, very, very high coverage genome sequencing to calculate the donor allele frequency at all these SNPs. This will give us a picture of the transformation frequency across the chromosome. This is that data. This is a pair of experiments where we selected for different markers to select for cells that were actually transformed. And this is 40,000 dots squished into a very small area. This was technically challenging from a bioinformatics perspective, but it actually does work quite well. We are able to measure transformation frequencies this way. The data are reproducible if you go away from those markers, and we are really seeing transformation and not sequencing errors. If you zoom in a little, it looks a little more interesting, so you can actually see some structure. There's a lot of peaks and valleys of transformation frequency. The median transformation frequency is just what we saw for those phenotypic markers, about 0.3%, and then there's ups and downs. If you look across the whole chromosome and stare at this for hours and hours, what you arrive at is that there's hundreds and hundreds of these strong recombination cold spots. And then there's these kind of regions of normal peaks of transformation, and then there's a very few number of very hot hotspots, which I'll mention in a second. We did some statistics to try to correlate the transformation frequency with things that we thought would matter. One of them was that uptake signal sequence that's so important for DNA uptake. We thought if you were closer to an uptake signal sequence, you'd have a higher probability of transformation. And we also thought that if you had more or less sequence identity, you would have more or less transformation. And those two things were true, but only weakly explained the result. I'm going to kind of move quickly through this. There's a lot of missing information that is pretty cool that I'm really interested in doing that we can do to try to understand what variation we're missing. It could have something to do with the structure of the chromosome itself, which would harken back to my PhD days, or it could be something about some combinations of alleles being put together in a cell killing it. This is really interesting, and we followed up on this a bit, which is there were 3 places in the genome that were extremely hot hotspots. So if the median was 0.3%, we were getting some places that were upwards of 8.5 to 9%. Exactly 3 places. Everything else was down more in the 1 to 2% area. All 3 of them were very large outer membrane proteins that were involved in the transport of heme iron. And notably, this one happens to be right next to a pair of competence regulated genes that form a toxin-antitoxin system. Another analysis that did an evolutionary analysis across many species looking for hotspots of recombination in bacteria actually also found one of these across many different species. So it looks like it's probably something real. Okay, I'm going to do this one real quick because I think I'm going for like 40 minutes or something like that. Is that true? I'm going to say it's true. I'm going to do a bait and switch first because until recently, I really only did experimental work and not so much comparative work. Oh, all right. And so this is going to be a little bit of a narrower question, which is, can we use transformation to identify pathogenesis factors? presumably, if this is happening out in nature, this is how they can transfer around. Maybe this would actually be something useful. This is right around the time when I'm starting to try to think about getting a job and staying in science, academic science. And I wanted to try to think about ways that I could use this to get a little bit beyond just studying transformation itself to something that might be more useful. This is where one of my conference buddies, Hunkal Garmendia, comes in, who at the time was extremely interested in intracellular invasion of lung epithelial cells because of the role of Haemophilus influenzae in chronic obstructive pulmonary disease and the likelihood that intracellular invasion is part of why these lower lung infections can be so persistent despite antibiotic treatment. There's other reasons too, but this is probably one. And this basically started from the premise that we checked some strains, and some strains were really good at intracellular invasion in a gentamicin protection assay. Some strains were terrible at it. So if we use the DNA from this strain as the donor to these, then perhaps we could map a gene. And so this worked quite nicely. Essentially what we did is we made a pool of recombinants and we fed them to airway cells, and then we selected for invaders, and we did that over and over again. So that what we were doing was enriching for those recombinants that were better at invasion. It happened real fast. We did 2 different recipients. They both rapidly reached levels of invasiveness nearly comparable to that of the donor. And if we didn't add DNA to those competent cells, then that didn't happen. Just as a note, I have another PhD student who finished just at the beginning of the pandemic who was going to do this same experiment. It's not always going to work. So he tried it with serum resistance and complement-mediated killing because there's so much variability in resistance and sensitivity, and it totally failed because he didn't have to add DNA to get any of the sensitive strains he tested to become resistant. And that had a lot to do with phase variable genes. And so that was the sort of trick. A lot of phase variable lipo-oligosaccharide modification enzymes was one of the main causes. So this one always worked, but it didn't end up working in this case. If we looked at sequenced pools of recombinants at these different cycles of selection, you could see that we enriched for some segment of this genome. When we looked more closely, we discovered that there was this whole operon inserted into this location. And it turned out to be a really, really well-studied protein called high molecular weight adhesin. I'll get to that in a little more in a second. So the insertion of this whole operon from our donor strain to our lab recipient strain induced this high invasiveness. And then our other recipient just did an allelic replacement of its HMW2 with the HMW1 of this from the donor. We did mutant confirmation that if we mutated the HMW1 in our recombinant, it turned into it looked like the recipient strain again. And then we submitted our paper, and we got this kind of problematic review, which said, so we know HMW1 is an adhesin. How do you know it's involved in invasion? Maybe it's just sticking to the cells better. And so we did the assay, and sure enough, like it's way better at adhering to airway cells. And then it got even worse because if you just leave the cultures lying around on the bench, the ones that are the donor strain or the recombinant, the cells just sink really fast. They self-aggregate, they auto-aggregate, and they just sink to the bottom of the culture really, really well. Whereas the recipient strain and the mutant remain well suspended after hours of sitting around. This is made us sad. We didn't really know what to do, but it turned out that the Joe St. Gem lab here in Pennsylvania, Philadelphia, know all about this gene, and we talked to them for a while, and they're like, you need to do some immunofluorescence. So we did some immunofluorescence. And what we discovered was this really kind of strange, interesting emergent property of this self-aggregation mediated by this one allele of HMW1 or 2, which caused the bacteria to seem to invade as groups. So we don't know these bacteria to divide intracellularly. These are airway epithelial cells. This is their nucleus. These red bits are these endosomal vesicles, and these yellow bits are bacteria. The bacteria that's been taken up and hanging out in this cell is usually seen as a single cell, but in this donor strain or in our recombinants, large clumps of these bacteria were presumably being taken up as a whole. So they were self-aggregating before they got there and then being taken up as an aggregate. So that was really the explanation for why they were invading better. It wasn't an invasion. It was something that allowed them to invade as groups. So this happened quickly. A lot of people do laboratory evolution experiments, but they usually do it just looking for mutations. And here we're just doing pretty much the same thing, but instead we're looking for natural variation from a clinical isolate with a pathogenic trait and figuring out what the actual genetic constitution of that is through a simple transformation experiment and sequencing. So there's other things we could do here, especially kind of in combination with what Danielle had done with her very, very large DNA transformation, where you could imagine we could build mapping populations like people do with plants and animals that you could reuse on almost any phenotypic trait. But it might not always work, like with Kevin's experience, where we have this highly evolvable trait where rapid mutations can cause something to flip from sensitive to resistant really easily. So this sort of gets us through this kind of transition from when I was going from my postdoc to looking for a job. And we answered some questions. We discovered that indeed they take up a lot of DNA. Some parts do recombine better than others for somewhat unknown reasons. And very rapidly we can see how selection can operate on recombinants to cause rapid adaptation. A little before this, how am I going to get a job? It's like so competitive. It's brutal out there. It's just really rough. I mean, the fact that I actually got a job came down to a lot of different serendipity. But one really kind of important piece of advice I had gotten is I was applying for a lot of jobs in like biology departments in universities. And one of my advisors this is his direct quote. He said, "Get thee to a medical schoolery," right? And the advice was really about what I did. It had to do with the organism I studied. He's like, no one's going to want you to come in there and teach like intro evolutionary biology. They're going to want you to teach like bacterial pathogenesis and stuff like that. So like you need to go where they want that. And you need to shift your gears and start thinking a lot more about what people want to do about this pathogen. And it's something I was maybe navel gazing a little too much, and I really needed to start doing that. And so since then, I've been trying to learn more about how my organism lives in the world and understand a little bit more about how I could be impactful in a public health context. And so I know this is getting late and we started a little late, so I'm going to do this hopefully quick. But I think we know already that this organism lives in mucosal surfaces and the airway. It likes to live in the upper airway in the nasopharynx. It has a terrible name. It does not cause the flu, and it does not love blood. It often dies in blood. But it used to kill a lot of babies. There's that vaccine against the type B capsule ended that. There's very, very few cases of type B Haemophilus influenza anymore. And now we mostly worry about it in the context of morbidity. So one context, I'll tell a very quick story about COPD and how this is mostly in the elderly, mostly former smokers. There's a lot of causes of COPD, multifactorial disease, but what it's characterized by bacterial infections of the lung and exacerbations that are often caused by Haemophilus influenzae. And the other context is in that of pediatric ear infections. This is very, 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 very common. Most kids have at least one ear infection. And it has a lot to do with where this organism lives as its normal niche, just next to the eustachian tube. And so eustachian tube dysfunction and various other things can lead to bacteria ending up in that middle ear space. So we come back to these big questions about how we can sort of use these various tools to like understand why these diseases happen or don't. I'll start with this example. A lot of this is published. We're still working on it. But essentially, the premise here is that people who get infected with COPD, who have COPD, have these pulmonary exacerbations, often caused by Haemophilus. You can feed those people lots of antibiotics and kill off their infections, and then 6 months later, they come back with the same infection. If we track these populations over time and watch them adapt to their new niche, can we see evidence of recurrent genomic change? And could that tell us about molecular pathways that are involved in how these bacteria go from being a nasopharyngeal commensal in a kid to infecting their grandpa's smoker's lung for the rest of their life? And we did this in collaboration with Hunkel, again, who had access to a collection from a clinician clinical microbiologist, Sarah Marti, and we sequenced this longitudinal collection of isolates taken from 13 different COPD patients. And what you can see here is a phylogenetic tree we produced of all these isolates, starting from the middle and working its way outward. And the little, each color, it corresponds to a set of strains that are very closely related. So these green ones are all closely related. These purple ones are all closely related. The black ones represent lineages we only saw once in this collection. And then here's how they lay out over time. So one thing to note about this that really distinguishes it from other opportunistic pathogens from the environment is when we look at those kinds of like CF pathogens, it's often like one clone that's evolving the whole time in one patient. But here, this is a normal human commensal. So there's a lot of diversity. When we get these isolates, they kind of, the lineages kind of come and go. And there's sets of bacteria that are similar. Maybe they get cleared after that, and maybe they get reinfected from their grandkid with a different strain, and so on. In any case, what we did with this is we found evidence of recurrent genetic change in the same genes over and over and over again. This is actually an outdated list from the publication. I don't know how I did that here, but this is effectively the same list as we saw in the paper. There's a bunch of genes where out of the 20 different clonal lineages that had more than one isolate, we saw the same gene changing over and over again. We really emphasized this one FAD-L at first because it had a really interesting outcome. FAD-L is a fatty acid transporter and an outer membrane protein that sticks to airway epithelial cells. So it's required for Haemophilus influenza to interact with this CCAM1 receptor on airway cells. And when it's deleted, adhesion and invasion and mouse lung infection all get worse. And so we were perplexed as to why this gene seems to be getting lost. All of the mutations that we saw were eliminating the gene's function entirely. And so it was really kind of inexplicable to us unless it was maybe an immune evasion thing. Maybe this FAD-L protein is really immunogenic. That didn't seem to be the case. But what we found that it was that in COPD lungs specifically, there's this high concentration of arachidonic acid, and the wild type bacteria dies because it takes up that arachidonic acid and can't do anything with it, and it ends up killing it. So the mutants fail to die in the COPD lung. So this is this sort of tension between one function giving the organism some help in its niche, but in this particular environment, it doesn't work. And so when we looked outside of lung infections, and we looked at all the other bacteria, all the other Haemophilus that had been sequenced, we found almost no instances of this gene being broken. And so this looks like a place where this gene loss that these bacteria are undergoing to adapt to the COPD lung is restricting them to now be pathogens. We've gone on to do more work in this system and with this collection. There was one really interesting one where HMW1 came up again because of phase variation, where the promoter of HMW1 started out quite short with expression of this HMW1 allelic variant that allowed hyperinvasion. But then as time went on, as we had serial isolates from this patient, the repeat in the promoter expanded and it downregulated expression of this gene and caused the bacteria to take on a more biofilm type of state. So that was sort of another outcome of that collection. And we're still working on this collection. And this is sort of where we're at now, where this is the same phylogenetic tree here on the left. The colors here represent the different subjects that the strains were taken from. And then this heat map represents phenotypic variation for various phenotypes. The biofilm level, the growth rate, self-aggregation, hydrogen peroxide resistance, serum resistance, and polymyxin E resistance. And what you can see is that across this collection, there's a great deal of variability. This is all normalized phenotypic data, but I assure you that like all of these, the actual range of these traits is quite broad. And one of the weird things that we can do here is we can just kind of look at it and find instances like here. These three isolates had different biofilm formation, and we can look at the genomes of those three very closely related isolates. And this is the entire set of genetic differences that distinguishes those three isolates. There's no others. They're very close together. And so we can almost pick out the individual mutations that must be involved in this much higher biofilm score for this strain. It must be this SNP, this frame shift SNP in a LOS gene, or this promoter deletion next to a gene of unknown function. So one of those two must be responsible for this huge shift in this biofilm score. There's other cases where we can't see that. So in these natural strains, there's also recombination that has happened. And so, for example, here we have a couple different phenotypes that vary across these set of very, very closely related isolates, but it's a little bit obscured by the history of past natural transformation that has happened. The last thing I'll say about the COPD story is that on these lists, all 3 recombination hotspots that I had detected in that natural transformation mapping experiment appear. And in all those cases, it looks like the reason the gene's changing so much is not because of point mutations or phase variation, but because of the entire allele being replaced by recombination. So this is further evidence that what we were detecting in those in vitro transformation experiments is happening out in the world. Go a couple more minutes, maybe. Let's see. I can do this quick. So this is what I'm working on the most these days is thinking about this issue of children's ear infections. The reason for that is that it's really common. And even though it doesn't cause any kind of death, the kids who experience chronic and recurrent ear infections, otitis media, can have some real long-term, lifelong consequences. One of the main ones is just that it's during toddlerhood, and if you have many ear infections, it's right when you're acquiring language, and so there can be delayed language acquisition and other issues. The kind of epidemiology of ear infections is also pretty interesting, it turns out. The one thing that's quite interesting is that the introduction of vaccines against the pneumococcus has actually shifted around the microbiomes of people's NP. And now as the pneumococcal vaccines improve, we're seeing less and less of the pneumococcus causing ear infections, a little bit more of this other one, Moraxella, and then the Haemophilus are staying about the same, but which strains are showing up and not showing up is changing. It's pretty cool. So how are we going to identify virulence genes involved in OM? Well, it's going to be easier than the last one. The last one was harder. It was COPD. What we had to do was everybody had COPD, and every isolate we had was from an exacerbation. We had no phenotype. That's why we did the phenotypic assays later. Here we had this ability to acquire strains from both health and OM disease states, even from the same kids. And then we sequenced their genomes and we analyzed them, and we found some virulence genes. And so the way this worked was essentially through a wonderful collaboration we have going with this guy, Michael Picicaro, up in Rochester General Hospital, who's been doing a very long-term survey of children longitudinally, where he enrolls them prospectively and samples their nasopharynx and nasal passages for bacteria over the first 3 years of their life. And if they ever have an ear infection, which plenty of them do, then they not only do the nasal workup, they also directly sample from the middle ear by tympanocentesis. And they do this amazing separation of different bacteria, as well as like the supernatants and pellets of the ear liquid, so they can do like cytokine profiling and so on. It's pretty cool. It's been going on for a long time. The y-axis is 1,200 subjects. The x-axis is time, and every pink spot is an ear infection. This is the same data, but showing all the Haemophilus influenzae in blue. And so we couldn't sequence all 2,000. We didn't have that kind of cache. So we picked out a set of bacteria from these 20 subjects. 9 of the subjects never really had very many ear infections, and none caused by Haemophilus influenzae. But you can see these filled circles. There were plenty of these kids were colonized by Haemophilus influenzae. These kids are a class that's studied by Michael Picicaro, who are prone to getting otitis media. This probably mostly has to do with slightly delayed innate immune development. They not only get more ear infections, they also get more other infections, and they also have a little bit low responses to vaccines. And you can see how many ear infections, every single green dot is an ear infection. And so some of these kids experience 12 ear infections before they're 3. So we got the isolates, we sequenced them. We used an Illumina. We also used a PacBio. Then we had to do something with the analysis. I'll blaze through that too. This is sort of like my way of thinking about it is before we can, we're going to do a simple approach. We're just going to treat all the genes like they're in a sack. But before we do that, first we have to assemble the genomes. So we assemble the genomes with various ways, and then we annotate the genomes so that we can get a hold of the genes, individual genes. We do this with various software suites, and also cluster all of the genes that were detected across all of the strains into clusters of orthologous groups. That's what's shown here. This is that same gene possession heat map I showed you before. After it was all said and done, we had about 250, a little over 200 genomes. Some of the genes were found in all strains. Some of the genes were found in only some. We then wanted to do an association test where we were going to find genes that were associated with disease or not disease. And so what we needed is a control with some type of phylogeny. We built the phylogeny from the core genes so that every strain had them, so we could concatenate them all together and feed them to a tree building program and make this tree. Again, these colors correspond to sets of isolates that are extremely closely related. And then this is how it lays out again over time. So here's our subjects again, and now I've colored each dot by what lineage it came from. And you see the same thing we saw in COPD. Strains kind of come and go. The fill means it was an ear infection. And now we want to just keep it simple and just look at whether, we're not going to worry about the core genes. We're just going to worry about genes that are present or absent. We're going to see if any of them are associated with disease. So these are the ones that we're going to look at. And we're going to look at it on the context of this tree. And that's, we can't just simply say are more of, if we look at one row, more of the blue are in disease and more of the white are not in disease. We have to control for this population structure that we have here. When we do that, we end up with this list. This is a very strict list. There's 6 genes that we found whose presence or absence was associated with whether we picked them out of a sick kid's ear or not. And there are 2 things that struck me. One was that I didn't know most of these words meant. And the other one was that the effect was negative. So it happened again. So 5 of the 6 genes that we found, it was their absence that was associated with disease. The gene's presence was associated with health. There was only this one that wasn't like that. Didn't know what the words were, except that I know that putrescine stinks and cadaverine stinks. I looked up what they are. They're called polyamines. I learned too much about them. I'll tell you a little bit more about this one. This is an operon. It's the POT-E-SPF operon. It's a putrescine ornithine antiporter and an ornithine carbamoyl transferase. And its absence is associated with isolation from the disease state, particularly from the middle ear. It's not just this weak association, it's also this phylogenetic association. If you look at this tree long enough and you count up the gene trait co-occurrences that have occurred, whether the gene is gained or lost, and whether that's associated with the tips being from disease or not, you can find at least 7 instances of gene trait anti-occurrence. What do these genes do? Well, in E. coli, it's well known because luckily we know E. coli well. The POT-E gene is expressed as a transporter that takes in the neutral polyamine ornithine, and the SPF converts ornithine into a weakly basic putrescine, and the POT-E then exports that. So what this allows E. coli to do is to survive in acid, not by surviving in acid, but by neutralizing the medium that it's in. I called Dr. Pekikaro. I'm like, so what's the pH of middle ear fluid? And he's like, it's alkaline. That's real weird, isn't it? And I'm like, I don't know. I'm not a doctor. And he said, it's really weird. It's really weird that we think of pus as usually being an acidic thing. And it's weird. We just found this out recently. And so now I'm trying to build these hypotheses about like, why would losing this operon that allows you to survive in an acidic condition be useful in the context of middle ear fluids that are often alkaline? Well, maybe it's kind of the opposite. If you have the system, you survive in acid, but you're like more sensitive in base. So that was sort of where we were at. I also call Hunkel, and I'm like, Hunkel, what do I do with this putrescine ornithine antiporter? And she's like, I made those mutants 8 years ago. Want them? She sends them to me in the mail because she didn't have any use for them anymore. She made the mutants. They made the mutants and they didn't do anything in their COPD mouse model, so they didn't care anymore and they put it in the freezer. So she sent us the strains, and then we had some other kind of nice experimental verification of the function of the gene and also its in vivo relevance. And I'll cook through that stuff. So we did some work to validate that the absence of this operon did affect the phenotype of the cells with respect to whether or not we added ornithine. So again, I don't know much about microbiology. It turns out if you grow a culture of bacteria, the media will get more acidic the next day because all the metabolic metabolic products are often acidic. And so the pH of bacterial cultures that start at some pH tends to go up in wild type. But if you knock out as a function of addition of ornithine, but if you knock out these genes, then ornithine doesn't matter and they don't change their pH. If we looked at the basic condition in a growth curve analysis, in the basic condition in wild type, at high ornithine, we would see this decrease in the biomass overnight. And we didn't see that in the mutant. We validated that had to do with viability. So the wild type dies when we add ornithine at high pH. And Joris Beld at Drexel verified for us that the mutants failed to take up and convert ornithine into putrescine. So here's our wild type, and here's our mutant. It was especially, if we add ornithine, we get very high putrescine levels, especially extracellularly, as we'd expect. And if we knocked them out, we get very little. So this is like, well, how come? What's going on? Who's inhibiting who? What's happening here? We had a final validation experiment from Ravinder Kaur, who's also up in Michael Pekikaro's group. And she's been using a mouse model of acute otitis media where they infect mice with the flu virus, wait a week, inoculate them with NTHI, and then they can look at the burden of bacteria in the nose, in the wash of the ear, and the middle ear itself. And what we find is that the double mutant has about 100-fold increased in bacterial burden, both in the middle ear wash and just in general. The colonization is higher and also associated with the middle ear bone itself. So this validates for us that the absence of these genes leads to much higher virulence in this mouse. The pH didn't do what we expected it to do. The pH actually went up in the mutant. This might just be an indicator of this being a host response, that the alkalinity produced in the middle ear fluid is from the host, and it's responding somehow to the presence of larger numbers of bacteria. It doesn't do this in COPD. This is a tiny effect. It's barely significant, and that's why Hunkal had stopped doing it. It's not the best lung model. The colonization is very low in this lung model, but the absence of pod ESPF doesn't do anything. So we think that the host is causing this change in pH, and we also think it's specific to Haemophilus influenza. This is the pH of middle ear fluids collected by the Pekikaro group from children that were only infected by one of the three main ear-infecting pathogens. And just to note, all of these samples were collected into buffer. And so yet you can still see a significant difference in the Haemophilus influenza pH values coming from that middle ear. Okay, and then I'm going to kind of blaze through this, but there was this other operon on the list that had another stinky chemical that was also a polyamine and was also involved in central metabolism. And I also didn't know that my organism doesn't have a full Krebs cycle, it has like a Krebs line. And I also didn't know that polyamines are born from this Krebs cycle, including putrescine. But also ornithine comes into this Krebs cycle from over here at Krebs line. And the products that produce that cadaverin also show up on our list coming off as lysine. And so there's this weird interaction with primary metabolism that I don't understand, except to know that when we look on our pot ESPF mutant, we affect cadaverin levels. So this is again from Joris, where cadaverin in the mutant is essentially eliminated. So that suggests that we can't really tell what the direct effect is. So I'm a little confused and I'm still working on it. But we can indeed confirm that this is a true anti-ILM gene. I'm going to end quickly and just say that this stuff works. Weirdly enough, the one positive association with the gene was something that we had just very recently finished up work from my postdoc, characterizing some unknown competence-regulated genes. And it turned out that this competence-regulated toxin-antitoxin system is somehow associated with ear infections. So another way that it wrapped back around. So I'm going to kind of leave it here. The anti-NTHI genomic diversity matters is true of all bacteria. I almost asked that question to everybody that was talking about diversity. I'm surprised to find that the absence of genes is the thing that's being associated with disease. It makes grants a little harder to write, right? If you're finding virulence genes, you're finding things you can target with drugs or therapies. If you're finding anti-virulence genes or genes whose absence is associated with disease, it's harder to figure out how to use that to invent a new biomedical treatment. And I think that we're onto something with this particular gene absence being associated with ear infections. It's something to do with pH. And we also have all these barely tapped resources for trying to understand these infections. I'm going to move on from here and just state that I all needed all these people, and I want to thank you for your attention and for staying late. And I'm happy to take any kinds of questions you have. Thank you.
Thank you so much, Josh. I'll give like a quick clap for everybody. I certainly have questions, but I did see some questions in the chat. Okay, so let me just read one. So the first question is, how do you know that several fragments from different regions don't co-transform simultaneously?
I think this must be referring to the data I was showing from Danielle, showing long distance co-transformation. And the reason is because of the co-transformation rates. Of course, we could have two independent fragments transform the same cell, and we know that that happens. In fact, when we use the markers on opposite ends of the chromosome, we know that's what's happening because none of our fragments are half a megabase large. And so that's sort of set as our control. And so the linkage that we see is for the intermediate length fragments of 250 kb. But to more directly answer the question, if we really wanted to prove that we were getting recombinants that replaced 10% of the chromosome, then we need to sequence clones. And we couldn't quite afford that because the rate at which we calculated we would see those very, very, very long recombination tracks, we would probably need to sequence several hundred clones to get one. And we weren't really ready to do that. We do have some other data that might let us address it like a little more directly, but it's a valid question for sure, because it's something that it would be nice to be able to drive it that direction for technical reasons, but we do have to be very careful about it. Thanks. You're mute, Kayla.
This long into the pandemic and I still struggle with the mute button. I was about to say that was for Dr. Shefferly. Okay, I think we have time for one final question from Dr. Tam. What are the evolutionary advantages and implications for hot/cold spots for transformation/recombination? Are these genes around cold spots less beneficial for gene transfer? Are they sequence or location dependent? If you change the sequence around those hot/cold spots, does it change the rate?
Excellent question. It's something that I'd really like to be able to invest some resources in. The evolutionary consequences of having variable recombination rates have been explored by like some theorists and stuff like that. And it certainly lends itself to thinking about like diversifying selection versus purifying selection. So for example, the fact that these big honking outer membrane channels that transport heme seem to be the three really, really hot hotspots that we see might not be a big surprise from the context of like immune pressure on these cells and the essentiality of these cells taking up heme. So the hotspots evolution at those locations could be being driven by that selection for diversifying selection. It could also be just something mechanistic. The one that I pointed out is right next to that competence regulated toxin antitoxin system. There could be sort of like the mechanistically, the hotspot could be arising from things like topological constraints on the DNA and so on. But why they would evolve in one place or another could very well be because of the function of the genes. The cold spots are a little harder for me to do much more than just baldly speculate, because I'm not totally convinced that the cold spots are truly recombination hotspots. They may be sites of genetic incompatibility between the strains I'm using. So they could be divergence between interacting subunits, and I can't confirm that because my experiment required me to select for the cells to be alive. And so I don't know whether I broke some kind of genetic compatibility with those at those cold spots. But there would be some predictions that purifying selection might be something that cold spots, it would be beneficial to be in a cold spot if you were undergoing high levels of purifying selection. And so you might predict essential genes to be concentrated in cold spots. Or you might think, or it could have nothing to do with that, and it could just have to do with the structure of the chromosome and like where the genes are located with respect to where the incoming DNA is. But I think those are really interesting questions. I would love to start moving the hotspots around and seeing if we can manipulate them and affect the rates, but I haven't been able to do that yet. So I'd love to know that answer. I would like to suspect it's something to do with topology of the DNA and to do with like nucleoid structure, but I have no evidence for that.
Thank you. So that was an awesome talk, and thank you to all of the speakers for today. It's not easy doing it for this long on Zoom, and we're still going strong, and I think we had a wonderful meeting. So thank you all, and I think that's it.
Thanks for your patience, everyone.
Oh, it was awesome, Josh.
Thanks,
Don't say that. It was
Oh, no,
awesome.
I just mean, sometimes you try to say too many things at once. Once you're like, oh, just give a retrospective of your kind of scientific path. I was like, oh God. And but yeah, thanks for everyone.
Thank you.
Good job, everyone. Thanks to all the speakers. Happy to be here.
Great job.
Yeah, wonderful session, everybody. Have a good night.
Have a good night. Okay. I always feel like, do I hop off? Okay, I'm good.